So we're going to call the afternoon to order, and just to confirm that we have all the CICATA members present, both in the room and online. Okay. So with that, we're going to move to the next item for discussion, which is the CICATA discussion for the developmental neurotoxicity section. Do the lead discussants have a particular order that they want to go in? Otherwise, I'll just go down the list. Okay. Sue, do you want to go ahead?
Yep. So let me start by just giving some thoughts on the DNT in vitro battery. I really think that it's some elegant science that's gone behind this to target these critical key events in neurodevelopment and then develop assays against these. And it's really challenging, right? Neurodevelopment is extremely complex. It's a multifaceted process, different localized effects, different timelines. So really difficult to be able to model and predict DNT. I think this is not going to be unusual going forward, because I think as we get to more and more complex endpoints, the NAMs that we need to develop are going to be coming in battery formats like this. So it's going to be important for us to learn how to manage these kinds of processes. So battery right now is 17 assays, and there are also still remaining some notable gaps in those assays. And so one of the things that's interesting is that even if you come through negative through all of the DNT in vitro battery endpoints, that doesn't mean that your compound is negative for DNT. So something to keep in mind. Also, the relationship between the assay outcomes. So something that's positive in a DNT assay, it's not clear at this point in time what the adverse effect is for that in vivo. So another thing, you know, it's going to be something that we're going to have to grapple with more and more as we move into these complex endpoints, I think. I want to commend the folks who work on this program, EPA, NIEHS, OECD folks. Really great job with data transparency throughout. So if you go in ToxCast, you can find the data. If you go in the ICE dashboard, the data are there. It's really, really been managed quite well. For activities in which NICEDM and ICMAM can be involved, some of these are ongoing. So, you know, just I guess for awareness, NIEHS is probably already engaged in many of these, as might be EPA, but just to level set. Standardized data management is a big one. So EFSA is already, I think, a proponent of the benchmark concentration approach. In order to make that work, you have to have a benchmark response, and those have typically been identified by the assay developers. And so usually they're picking a point where they think they see a meaningful assay change. They're considering variability in the assay as well, but also trying to limit false positive results. And is the BMC the right way to go? Perhaps, or perhaps not. But it would be really great to see a consensus approach come out of this program on how to manage those data. Some other really helpful things would be defining performance criteria for the DNT assays. So positives and negatives that should be used in the assays, as well as acceptable variability in those assay endpoints. A regulatory decision framework. So how many positive assays or endpoints are needed, or at what potency to have a compound move forward in the DNT assessment, right? So there's 17 assays. You think about an assay like MEA, there's maybe 14 endpoints in the assay. Those can be increased or decreased. If you see a change in one of those 14 endpoints within one of the 17 assays, is that important to drive forward? And it's really, you know, something to think about when we see like saccharin and, you know, ascorbic acid also have some hits in the assay battery. So something to think about, right? There's no good consensus right now on battery reference chemicals, right? So there's been a new publication that came out of the EPA on 10 favorable negative reference chemicals. And so that will be an opportunity for us to move forward and look at those. But without having agreed upon reference compounds, it's really difficult to gauge the accuracy of the battery. So something else I think that would be helpful. Communication and collaboration is going to be key going forward, because trying to reach some consensus for you know, a global program like this might be kind of a challenge. So just something else to keep in mind. How can insights from the IATA case studies be used to develop more robust and predictive approaches for the DNT? I just wanted to highlight a couple of opportunities that I noted when looking at the DNT case studies. Would be helpful to have the development of additional DNT related AOPs. I think those are useful and help with the IATA approaches. I think training and education of all stakeholders, including regulators, on how to look at data that comes out of the DNT in vitro battery is going to be really important. And then expanding the chemical domain. So we saw that there's 81 chemicals that have been run through all 17 of those assays, largely focused on pesticides and other actives that have been identified, certain classes of industrial chemicals, but really it would be great to expand the domain to look at a wider range of chemistries that carry different physical chemical properties so that we can start to understand the performance of the assays in the battery better. So what criteria or methodologies might be needed to accelerate the use of data from the DNT in vitro battery? So development of fit-for-purpose validation, or whatever you may choose to call it, of complementary or orthogonal assays or more complex models, right? So as we think about getting positive hits in the DNT in vitro battery, what's our next step? Do we go in vivo to a DNT study? That's going to be a lot of DNT studies. Can we look at what that intermediate step should be? Are there other assays we can look at where we should see commonality in response using, complementary or orthogonal assays? What about zebrafish or brain spheroids or organoids? Can we go to something like that and be able to confirm that, in fact, that compound warrants a DNT study, so that we can make sure that when we're using animals, we're using them wisely? Other methodologies, there's lots of gaps that have been identified already in the DNT in vitro battery, and those are pretty much given in a number of the documents that are available. I just want to do a plug for the alternative models. The zebrafish, the planaria, the C. elegans, I think that those are really helpful models because in some cases they have metabolic competence, and in addition they have functional endpoints, right? So we get to see how the nervous system is actually contributing to function. What we've learned with a lot of the work that's ongoing, especially like in the zebrafish, I'll use that as an example, is that many of the methodologies are not standardized. So it would be really important to get to standardized methodologies to make sure that we have some consistency between how those assays are being run. Perhaps programs like CZIT, which is the zebrafish program that's already ongoing, will be able to help in some regards with this. Metabolic competence in the DNT in vitro assays, not very many of them I think have metabolic competence, so it would be helpful to be able to look at whether or not we have active metabolites being formed. And then in the QIV-IVE space, there's work, we have models that are including placental transfer. There's work going on in the blood-brain barrier. Lactational transfer would be another one that I think is important because neurodevelopment does continue after birth, so that would be important. Okay, and then the last topic area is factors to facilitate transfer. This was a challenging one for me because 17 assays, not a lot of laboratories want to take on, validating 17 different assays. The later stage assays like the MEA do have the opportunity that, you're looking at the convergence of many key events, so you might have an opportunity to pick up more substances, but there are also some of the more complex endpoints too, I think, that are measured. Some of the assays use specialized equipment, so we saw some of the data on high content imaging. So that's another thing that labs will need to invest in. And some of the assay materials, like we heard this morning, cells are not readily available all the time for groups to use to try and bring these assays in-house. And then honestly, for CROs are, they bring these assays in and develop them in these batteries like this. And this is what we saw with the EDSP program when those assays came in. They would like to know when people are going to start requesting those assays, right? It's really hard for them to bring them in and validate these assays, and then nobody wants to use them until 10 years down the road, right? So for them, understanding a timeline of when people are going to come to them to get those assays run really help to, get their interest to bring those in-house because there's a business model that works for that, right? Some points that might help to get other labs to bring this on. Standardized data management practices would be great. defined lists of positives and negatives, both for assays and the battery as a whole. And then we heard yesterday too, funding. So those are my, thank you.
Great. And we will have the next lead discussant, Dr. Sura. Thank you, Chair. I concur with a lot of what Sue said, so just I won't try to say the same thing, but just some of the things. First and foremost, I want to applaud everybody. Again, brain and developmental neurotoxicity is very, very complex. It's like you can't even think about unraveling one at a time, because one thing goes from the start, you start to do the other factors. So very, very complex. So really appreciate it. So, but looking ahead, like there are, and again, a lot of the gaps you guys have already identified. You know where you're headed and what are the gaps. And nothing that I would say would add a lot more, but maybe some things to consider would be like, I know we brought up the exposure component, and I think that is pivotal, because chemical exposure alone is not what is driving all the effects that we're seeing in humans. So we need to bring that in. And again, I don't have any thoughts, maybe looking at epidemiological data, clinical data, and animal studies. And again, what you have to have these studies which are well curated, and you have actually information that you can tie down to, because end of the day, it boils down to what is the dose, the concentration, and the exposure. So that is essential for understanding the developmental neurotoxicity. Additionally, like we are looking at the assays which are looking, currently the way I see it is that the focus on the middle bin, which is the structural aspect. We're not looking at the molecular initiating effects and the downstream effects, and everything needs to tie in together. And we should have a battery which captures the whole spectrum. I know it's not an easy task, so this is in no way or form saying that it is easy, you can get it done. It'll take years of effort, and I guess you guys are in the right direction, but definitely that is something which is needed. And again, mechanistic information is very important. Again, understanding what is happening at the molecular, cellular, and the behavioral level, because that is what we are seeing when we see these autism spectrum and all that. We talk about these, but when you talk about it, a lot of researchers are working on it, and they really do not understand the underlying mechanism. So very close collaboration with those folks, so that you're working in tandem, trying to identify, because they probably have seen an apical effect that we are not getting captured by these assays. I think that is very important. Again, looking for early indicators, maybe potential development of biomarkers, targets for the neurotoxicants, like the signaling pathways, because industries for the screening assays that is commonly used, we use a receptor binding and all those aspects. And I think that is a critical piece. And then again, coming back to the cholesterol example, the brain is relying on de novo cholesterol synthesis. So anything impairing that is going to change the dynamics over there as well. So that is another thing that we need to take into account. And last, but again, everything has been done, and you guys are doing it again. You're fostering a lot of close collaboration, bringing in the academia, as was evidenced by the talk about Newest Rosetta. It seems very promising, and it kind of seems like, 96 well, you can do it all. But again, it has to be vetted and validated. And again, last but not the least, again, you guys have talked about it. We need to have a tiered testing strategy. Skin sensitization is an example. We've done it. It's a success. So let's move it to more complex ones. But I know it's not an easy task. That's all I have. Thank you. Thank you. Dr. Miles?
All right. Thank you. So this is the joy of going last. I'm certainly going to repeat a lot of what Drs. Marty and Sarah just said, but I just wanted to first thank all of the presenters this morning for sharing the amazing work that's taking place in each of your respective agencies in the spirit of advancing and promoting the use of developmental neurotoxicity NAMs. We all recognize the value in having them and continuing to advocate their use. And it's certainly going to take a domestic and a global commitment to continue to invest in these critical tools so they may be used in applied to risk management and harmonized decision-making to help protect people as well as the environment in which we live. I'm encouraged and impressed, as I'm sure others are, to see how much progress has been made. And It's also exciting to be a part of the intensive scientific collaboration that's going on based on the shared value of developing these critical tools. So the overall goal of reducing the dependence on in vivo-based testing for safety assessment purposes is evident. In forums such as this, conferences, training opportunities that allow all stakeholders to participate and engage in thoughtful communication, help us build stronger relationships and inspire one another to continue to pursue innovative ways to advance the science, protect communities and the environment, have definitely been recognized. And I personally am very optimistic about the wider global regulatory acceptance and promotion of these tools. And we'll continue to work and develop. And we see how well they are performing and in many instances outperform some of the in vivo traditional-based models. With the greater use and promotion of these types of tools, I see this leading to increased efficiency in regulatory decision-making, which allows stakeholders to prioritize resources to focus on substances that show the highest potential for developmental neurotoxicity. And the foundation work that's occurred looks very promising, but it will be equally interesting to see how complex mixtures perform and compare in these assays. And it will also be interesting to see how these NAMs will be adopted across different industry sectors. We've seen a diverse array of chemicals represented this morning, so pesticides and flame retardants, but there is and will continue to be a need to explore other categories of substances in the future. Also highlighted this morning in the presentations, developmental neurotoxicity is extremely complex and involves an equally, if not more complex exposure scenario. And Dr. Marty touched on this as well, but maternal exposure to substances normally found in the diet and environment obviously are important. But I think we should also keep in mind about some of the non-chemical factors that may have a significant impact. And these should be kept in mind as well. So for example, how does stress involve the biological or impact the biological response or responses observed in these 2D, 3D models, and even in the zebrafish model? Stress could be in the form of increased temperature, malnourishment, or other associated conditions that are relevant to humans. Additionally, it would be interesting to understand if any of the responses are reversible, and if so, what are those mechanisms involved, and are they shared amongst certain substances? And lastly, this may be out of scope for this group's focus, but is there an opportunity to use these tools to identify maybe neuroprotective substances with the same types of follow-up questions? So again, thank you all for your collective contribution to advancing these tools, and I'm certainly eager to hear thoughts from Drs. Manga, Holberg, Schaefer, McPherson, and Colbrus. Thoughts on any of the questions and comments we've posed?
I will just add on to that as well, because not being a DNT expert, so I'm going to force one of you to come and answer my question. With the thought on complex mixtures, which of the battery are amenable to actually test with complex mixtures, if any?
Hochberg, nice to see them. So we can come back to this question when the phase 2 chemicals are coming in, because we are doing some mixtures that Chris was talking about. So those mixtures are not necessarily built on what you are exposed to as a human necessarily, but we are using complex mixtures with chemicals that have different mode of action. So I think there's, for pyrifos is included in there. I think it's metal mercury made with lead. There's some of those. So it's different modes of actions that we have different kind of mixtures, and we will be able to come back and answer that question afterwards. For the phase 3, we're also talking about looking at more relevant mixtures. So we're looking at mixtures from Superfund sites. And I know NCAT has already been starting to do some mixture exploring there. So we're going to use some of those mixtures also in the future lithium battery. So that's what I can say for now. Anyone else want to add?
Yeah, thanks, Helen. I think that's exactly right. We're going to have some mixtures data with the phase 2 and phase 3 sets as that data comes in. More generally, to answer the question, these assays are all, with the exception of the MEA assay, 96 well or greater plate assays. And so if there's a specific mixtures question about additivity or something like that, they're actually quite amenable to designing, well-designed mixture studies to address questions like that. They just haven't been utilized in that way to date. So because you have a 96 well plate design, you can do kind of array design for mixture studies where you can really get at, statistical models that address additivity or lack of, or other than additivity type responses. So I'll stop with that.
Thank you, Tim. Just as a reminder for everyone in the room, if they want to announce who they are before speaking. Megan?
I'm Megan Colbert. Can you hear me? Hello. Okay, I'm Megan Colbert. I just wanted to so I'm not going to comment on the data itself, but we have assessed heavy metal mixtures in the 4 assays that we've now transferred. We have a unique opportunity because we tested them as single compounds as well as mixtures. But I think what the real difficulty is not necessarily that they can't be assessed in the assays, it's how to model it, which I think is the difficulty for the mixture community as a whole. So it's great for us because we have single compound data, but if you don't for a mixture, then it makes it a lot more difficult.
Okay, if I want to open it up to any other Takata members that want to participate in the discussion, and why don't we start with Dr. Berg?
Thanks. It was a great presentation. So in looking at, how were the battery assays selected? It was presented, or I took from the presentations, that they were covering important neurodevelopmental processes. And so I'm wondering, how does that map really onto key characteristics of neurotoxicants? And because there's a lot of biology, but there tend to be these particular failure modes, sensitive failure modes, that and one would ideally like to design single assays or small numbers for those failure nodes and get most of the benefit with smaller study. That's kind of the idea. So that's a question I had is I know that there's been efforts in the past to do this with, key characteristics and those sorts of things, but mapping, that would help on the mechanism, because I noticed that all your, the rotenone and the methylmercury, they lit up all the assays, right? So to me, it's like, ooh, redundancy. I don't want redundancy. In a screening platform, you want to make it as efficient as possible. So that's one thing. And maybe just one technical point, I would really like to see confidence intervals around your measurements, because you've got a lot of significant figures there that have a confidence interval. So I think that would speak to what you said about a little bit about the good practices on your data.
Dr. Schaefer, was yours a response?
Yeah, especially with respect to the question about key characteristics. So first of all, yes, you're correct. The assays were designed to address these key neurodevelopmental processes. So there's not been a publication yet on the key characteristics for neurotoxicity, but that was a process that was started a number of years ago. It kind of got delayed and derailed by COVID. We are making efforts to get that key characteristics paper out. And I think when that does come out, what you're going to find is that there is a lot of overlap between the the key events that are being covered by the assays that are part of the DNTIVB, and the key characteristics for neurotoxicants that are going to be identified by that key characteristics paper. So that work is ongoing. It's a different group of people with a little bit of overlap between the assay developers, but hopefully there will be something out on the key characteristics within the next year. I'm crossing my fingers on that. So hopefully that helps to address that question.
Thank you, Tim. And Dr. Bingham?
Can you hear me? Yes. Okay. So this is Campbell Zeno from University of Pittsburgh. I have three questions, comments about the presentation. The first one is that my impression from the first three talks, at least by Drs. Magnus, Hogberg, and Schaefer, a lot of approach was retrospective analysis of the literature, which means what has been done, let's understand it, let's try to take it. Is it worth to consider a prospective approach, which requires a more closer tie with technology method developers? Because then perhaps identifying the niche or unmet needs or pressing needs would help to kind of capture those and work closely with the method developers. So that was my first comment. The second one is that it's a very broad comment. What do we mean by NAMs? Do we mean anything that is non-animal human relevant, or we want to underline the new aspect of this as well? Are we going to press for things evolving so quickly that we can integrate them? And by that, I mean anything from 3D bioprinting, organs and chips, deep learning, or we mean assays, which are non-animal, human relevant, like 2D models, cell lines, been around for so long. And now we use them for whether toxicology application, etc. Are we going to draw a line between the NAMs versus alternative methods, or we want to emphasize NAMs by kind of bringing the emerging technologies into the field? And my last comment and question is that, so I'm not a neuroscientist, so I see this from a pulmonary kind of expert, immunologist. There were so many endpoints that was important, from proliferation, apoptosis, migration, outgrowth of the neuronal cells. Does it make sense to, rather than having so many endpoints and then so many assays, to develop a battery of in vitro platforms to prioritize the most pressing ones that is relevant clinically, and then have multiple assays for individual endpoints, rather than having so many endpoints with one or maybe two assays to develop? So that's my comments and questions.
Thanks, Dr. Benham. I am Nicole Kleinschwer. Thanks, Catherine. Just to respond to the NAMS question, this is an acronym that has been tossed around by a lot of different groups and has many different definitions. As far as ICVAM is concerned, the definition that we have agreed upon, the definition that's in the validation guidance document and in the ICVAM strategic roadmap, is a new approach methodology, a broadly descriptive reference to any technology, methodology, approach, or combination thereof that can be used to provide information on chemical hazard and risk assessment, and that supports replacement, reduction, or refinement of animal use. So it is a much broader term. It does not stand for non-animal methods. It, I think, to your point, does emphasize the sort of the novel aspect in terms of providing insight that is mechanistically informative, mechanistically relevant, and supports moving the science forward in a way that improves public health protection and improves human and biological relevance.
Yeah, John Gordon of CPSC. I'll start off saying this is my personal views on this. Nicole's absolutely right. That is the definition. I've never been a fan of new, the word new, for a couple of reasons. One, there's a lot of these assays that are 20, 30 years old that are fantastic. So that's not new. Second problem I have is I have worked in industry. You say the word new to somebody in regulatory affairs, they say no. So it kind of, I don't like the idea of calling the whole science that, because that's my personal point of view. I don't mean to be slamming anybody or anything. But when you start off the conversation with new and no, kind of shooting ourselves in the foot.
Okay, any other Cicata members that
I just want to remind folks that while a lot of our agencies, Jen Good, FDA, a lot of our agencies are looking for human relevant in vitro alternatives, there are some agencies that regulate other species and also use NAMs. And so we're not always looking for human relevant.
Any other Cicata members that want to participate in the discussion for this section? Billy, right at the last minute. Go ahead.
Well, only because I'm curious what the other term would be. We don't say new. I know some people say novel. Is that a fair question to ask? Like, what is there a reasonable term?
Well, for a long time we used alternative methods, but that applies to a lot of different things now outside of biological, to mechanical engineering. So they've adopted alternative testing. But the name of ICFAM is alternative testing methods, not NAMs.
Okay, so we are going to move on to the next topic, which is computational resources. Just as a reminder, we will open it up for clarifying questions for ICFAM and Cicata members after each talk. But we're going to hold all of the discussion until the discussion portion of the session, which is at the end. So I'd like to welcome our first speaker. We do have a change in the agenda. The next speaker will be Dr. Nicole Kleinschreier, who will talk on NICETAM's integrated chemical environment, or ICE.
Thank you, Chair. So unfortunately, Brad came down with COVID. So we all hope he feels better soon, and very understandable why he could not be here with us today. And thank you, Brad, for staying home. So this is our annual update on ICE, the Integrated Chemical Environment, which I hope most of you are very familiar with, but I will provide a refresher, and somebody could click on the screen so that the clicker will work. That would be great. Thank you. So we did just release a new version of ICE just a couple of weeks ago. So we had a major release, 4.1. I'm going to talk through that, but I am going to sort of provide a reminder of what ICE is and what it contains, how it's being used, some updates to the data sets in ICE, updates to the tools, and some strategic planning and activities that we have engaged in recently to try and get external feedback on ICE and continue to improve ICE, and some work that we have ongoing and future enhancements. And I should say that a lot of what I'm going to be talking about today has been inspired over the last several years by feedback from this committee. So we do have the computational tools section as a standing agenda item every year. As you know, we do deep dives into one or two other scientific topics, but we always have this computational tools section because it is such an important part of our portfolio, and it is the way that we deliver high quality data and computational workflows to the scientific community, and we work to make those as useful and broadly applicable as possible. And I'm extremely grateful for the feedback that we've gotten from SACADM over the years, which I think has really helped us to continue to improve ICE and meet all of our goals there. Excuse me. Okay, so what is ICE? The integrated chemical environment, it contains reference data, so high quality, curated, legacy in vivo data, and also in vitro data. So that's not just in vitro high throughput screening data. Oh, wonderful, there's timings on this. Brad obviously speaks faster than me, if that's possible. Is that possible? I don't think so. So in vitro data from the curated high throughput screening data sets, also from mechanistic lower throughput in vitro test data. We have in silico toxicity predictions from the OPERA suite. So Kamel Mansouri, our lead computational chemist, who will be speaking next about a new project that we're launching. He develops and maintains a suite of quantitative structure activity or quantitative structure property prediction models. We also have reference chemical lists and chemical quick lists, so lists that can be used for validation of new methods, or just lists that are of interest to the community, such as the Tox21 library, or pesticide active ingredients, or most commonly used inerts, or chemicals that have been run through NTP cancer bioassays, or that have been considered by IARC or the report on carcinogens. And then we have a suite of computational tools and workflows that allow for things like chemical characterization, predicting toxicity, but also doing computational toxicology analyses like in vitro to in vivo extrapolation, or running physiologically based pharmacokinetic models, looking at presence of compounds in various consumer products, overlaying exposure data. All of this is really in an effort to democratize access to these resources that were sort of previously the domain of subject matter experts. So now you don't necessarily have to be able to read or write code or be a PBPK modeling expert to actually run virtual animal models, virtual human studies, extrapolate from in vitro activity concentrations, and understand how they relate to doses or exposures. And many of you know the FAIR acronym. All of the data and data objects within ICE strive to adhere to the FAIR principles so that they're findable, accessible, interoperable, and reusable. Okay, so how is ICE being used? This is a nonlinear graph. Basically, the dark red across the U.S. is 15,000 user sessions over the span of a year, and then everything else kind of falls out after that, Canada being the next highest use case, and then the UK, Europe, China, Asia, Australasia, Brazil, and Latin America. We have quite a few users there. So over 27 countries, over 19,000 user sessions in the year that we looked at. So we're excited to see that ICE is not only being used by us and our federal agency partners, but also increasingly being used worldwide to support both research activities and regulatory decision frameworks. So here are just a few examples. We're actually investigating how we can best track usage of ICE in the scientific literature. So far, that really looks like me looking at LinkedIn and saying, oh, hey, look, there's another paper that got published that relied on ICE and used the ICE workflows, and sending it out to the team and saying, hey, guys, look, this is great. We didn't even know that this group was using ICE. They didn't even ask us for help. So apparently it's intuitive enough that these other groups are able to pick it up and use it. And this ranges from applying machine learning models and pulling data from ICE to really pulling computational toxicology workflows and using that to bridge things like human biomonitoring data and effects seen in vitro assays, to the IATA approaches that Helena presented earlier today. And then we also have a number of publications that we have put out to try and help guide the community and sort of provide guidance and details on how to use some of the interfaces and the tools that are in ICE. So what is currently in ICE? Over 5.5 million data points of just experimental data. So that's divided between in vitro mechanistic data and also legacy in vivo studies that we have carefully curated using both automated and manual curation pipelines. That total does not include all of the in silico predictions. So if you include all of the in silico QSAR model predictions from OPERA that have been run on the entire DSS Tox database, that's an additional 50 million data points. And those are predictions for things like toxicity endpoints, like endocrine disruption or acute systemic toxicity, chemical property predictions like physicochemical properties or environmental fate or ADME parameters, and a variety of other endpoints that we're continuing to develop out under Kamel's leadership. Here's more of a detailed view of the different in vivo and in vitro data that you can find in ICE divided by toxicity endpoint. And one thing that I really want to highlight here is that we spend a lot of time trying to annotate this data using knowledge organization systems. And previously we were using the NCI Metathesaurus, but to better align with some of the rest of the scientific community, we've pivoted towards using the OBO Foundry ontologies, which have much wider uptake. We still have linkages to the NCI Metathesaurus that are downloadable, but in version 4.1 that was just released a few weeks ago, the curated high throughput screening data, so all of the targets of these different in vitro assays are now annotated using OBO Foundry ontologies. Also in this newest release, we have added versioning information to the data sets. And we also have the ability to link to the National Toxicology Program and Division of Translational Toxicology STEBS database, so that's the Chemical Effects in Biological Systems database, and the EPA Complex Chemicals dashboard in our download files. So previously you were able to do that in the table that you would get on the results page of ICE. You could link, you could click on the chemical identifiers and that would take you to that substance page on STEBS or on the dashboard. But we've now added that functionality to the actual Excel files that you download from ICE when you look at those results. As I mentioned, we also have in silico models and integrated approaches to testing that are included in ICE. So all of the predictions from the models that you see listed here, most of these are from OPERA, but some are also integrated models of in vitro assays that were developed and validated in support of the US EPA's endocrine disruptor screening program. So that includes like the estrogen and the androgen receptor pathway models that you see listed there. And then we also have exposure predictions. So we worked with the US EPA, John Wambaugh and his team and the ExpoCast project to bring in the systematic empirical evaluation of models, or the SEAM-3 exposure predictions for almost 500,000 chemicals, and those are also available in ICE. New in 4.1, we have included applicability domain information for the OPERA predictions. That was always an output of the OPERA software itself, but wasn't previously included in ICE. That is now included in the ICE graphical user interface. We also have a REST API function. So if you don't want to interact with the pretty graphical user interface and you would rather just programmatically access all of the data in our database, you can do that through the REST API, and you also will now be able to look at applicability domain for those OPERA predictions via the REST API. As I mentioned, we have these chemical quick lists that are part of ICE. They are loosely divided into reference chemical lists. So those are chemicals that have been used to validate assays. So they are extremely well vetted. They have very robust data associated with them. Usually they are also associated with things like internationally harmonized OECD test guidelines or test guidelines from individual regulatory authorities like the EPA. And so you can see the reference chemical list that we have for various endpoints on the left-hand side. And then we also have what we call our chemical quick lists, which are non-reference chemical lists. And I already mentioned several of these. There's a couple that are new in the version 4.1 release. So the PFAS quick list was one that was requested by a number of different stakeholders. We have a relatively new ICFAM work group that was formed out of a request from the White House Office of Science, Technology, and Policy. It's called the ICFAM NAMS for PFAS work group that is sort of writing a state of the science white paper on PFAS as a broad chemical class and what NAMS may be amenable to testing this very challenging and relatively diverse chemical class, and what's needed and where are there opportunities in that space. And so part of the activities of that work group involved also nominating this PFAS quick list to be included in ICE. And we did get this through the EPA. So don't be like me. When I looked at the new release of ICE, I immediately scrolled down to P and I was like, oh, PFAS, it's not in there. Where is it? Called EPA PFAS Master List. We also updated our ROC, so that's the Report on Carcinogens classifications quick list. So that's all of the compounds that have been considered and included by the Report on Carcinogens office, and that's based on the newest release, the 15th Report on Carcinogens that came out recently. So I'm going to walk you through some of the ICE tools and highlight some of the new additions to these tools. Just as a reminder, they are search, so that's the interface to our database, chemical quest, which allows you to look at chemicals that might be data poor and identify similar chemicals that might be more data rich. Chemical characterization allows you to look at various chemical properties, compare two different lists with one another, look at use categories, look at presence in consumer products. Curve Surfer is a way to directly interact with the concentration response graphs from the high throughput screening data. So again, we have a curated version of the ToxCast and the Tox21 data that incorporates analytical chemistry information and mechanistic target mapping of all of those high throughput screening assays. So in Curve Surfer, you can actually overlay various concentration response curves on top of one another and search by things like mechanistic target groupings. You can do that for single chemicals or multiple chemicals at a time. And then the PBPK and IVIVE workflows, I think, are some of our most broadly used tools that allow you to apply forward dosimetry or reverse dosimetry modeling, respectively, to do things like run virtual animal studies or even virtual human studies and say, well, if I was exposed to this much chemical, what would be my chemical concentrations in the body, in the plasma, in specific tissues? You can look at steady state, you can look at time course. And then IVIVE allows you to do the reverse. So say, if I had activity concentrations in these in vitro assays, how much would I need to be exposed to before I achieved internal concentrations that correspond to the activity that I'm seeing in these in vitro assays? So I'm going to walk through each of those tools briefly. So the search function, again, is the way that you interact with the ICE database. You have buttons where you can either select chemicals from our quick list, so our reference lists, and use those to populate, or you can just populate user-defined chemical identifiers, and those range from names. You can use DTXS IDs, you can use CAS numbers. There's a variety of different identifiers that you can now use, and you can mix up a whole bunch of different types of identifiers, and the system will automatically go into our backend, into our database, and find the correct synonyms and the accepted names and pull the data for those compounds. And then you can click on the Select Datasets button to select assays, and those data sets are organized by toxicological endpoint. So you can see all the tabs across the top of that Select Dataset function. So it's the curated high throughput screening data. Again, that's mapped to mechanistic target groupings to provide biological context to potentially naive users who may not be as familiar with those assays. And then other endpoints like acute lethality, sensitization, irritation and corrosion, endocrine, cancer, cardiotox, DART, and then we have chemical properties and exposure data and exposure predictions as a data set tab. Once you get your results, it's an interactive table, so you can download those results as a static Excel file. or the tab delimited text file, et cetera. But also, if you've searched for a list of compounds, but you want to hone in on the details for one particular substance, you can click on that structure and you'll get a pop-up view that shows you the data results for that specific substance. You can also link to the Chemical Effects in Biological Systems, or CEBS database, so you can go to that specific compound page to see what studies the National Toxicology Program has run on that substance and what those treatment-related effects are. That's all in sort of free text summaries on the CEBS page. And then you can also link to the EPA's ComTox dashboard page for that particular substance. You can filter the results by various columns and endpoints. And then once you've filtered those results, you can actually send those results to other ICE tools and other workflows. So then we have our Chemical Quest tool, which allows you, as I mentioned, to identify similar structures. So you may be starting with a list of data poor compounds, and you want to look for compounds that look similar, that might be more data rich, to help inform your analyses. And so you can upload those chemicals, or you can actually use the chemical drawing tool to draw those structures or input smiles. And then you can search and you can filter your results based on sort of Tanimoto similarity or other similarity metrics to refine the number of similar structures that you get. And you can do this for single chemicals, or you can do it for lists of chemicals. And then you can keep digging down into that data. And then you can also look at specific substructures that might be affecting those toxicological endpoints that you care about. And those substructures are highlighted for easy identification. Moving into the chemical characterization tool, as I mentioned, this allows you to compare different lists of chemicals. So perhaps you have a list of potential test substances that you're thinking about running through an assay, and you want to compare that list of substances to perhaps the list that was used to validate that assay, and see how different they are from the perspective of physicochemical property distributions, or where they fall within the applicability domain of that assay. You can compare those two different lists to one another. This is also the place where you can investigate where chemicals appear in consumer products. So we've partnered again with the EPA and have a full instance of the chemicals and products database on our back end. And you can, these are these interactive bubble graphs. So it'll show you product use categories, and then you can drill down into each of those product use categories and get into more and more detailed categories of consumer products and where chemicals may be found. And then you can also compare those to in vitro bioactivity profiles side by side to understand how bioactivity may be related to exposure for the chemicals of concern. Again, Curve Surfer allows you to interact with that high throughput screening data. It allows you to overlay curves for various chemicals or various targets. One of the things I love about Curve Surfer and about many of our other tools is this data-driven filter chain. So if you are looking at a whole bunch of data across a lot of different assays for a large list of chemicals, but you really want to get to, okay, I care about endocrine-related targets, you can go in and you can filter on mechanistic target. Or you can say, actually, I really only want to see compounds that are active in the sub-micromolar region. And so what you'll get is this pop-up that shows you a kernel density plot of the distribution of the activity concentrations across the assays and chemicals that you've selected. So rather than applying an arbitrary filter, you can take a data-driven approach to choosing your filter and saying, oh, I see this distribution of activity concentrations shows me that, you know, there's a strong sort of bimodal distribution, and so I'm going to set my activity concentration filter at 5 micromolar or whatnot. Most recently in 4.1, we have added additional information regarding data flags on the results. We call them result cards. So every time you see a concentration response plot, it has a card associated with it. And so that not only includes like the mechanistic target information and the AC50 and the hit calls, but now it also includes things like potential technology interference. So we have both data and models to predict things like luciferase inhibition or autofluorescence. And then it also includes the data flags around things like analytical QC information associated with the sample that was being tested. And then the PBPK analysis tool is a forward dosimetry modeling approach that, again, you'll see a theme here. We partner very closely with the US EPA Office of Research and Development and help to, create interfaces that allow more widespread uptake and utilization of some of the tools that they have developed. And so one of those is the high throughput toxicokinetics R package. And so there's 3 different types of PBPK models that you can access from the HTTK package via the graphical user interface in ICE through the PBPK workflow. And so those are simple 1 compartment steady state models, more complex PBTK models. We also have a PBTK model that includes a fetal compartment, others that include things like inhalation and gas exposures. And so the user goes in and they just choose various parameters, the species, human or rat, the dosing route, the dosing length, where the ADME parameters should come from. Should they be experimental values only or predicted values, the exposure, the sort of simulation length, et cetera. And then you can run virtual studies in a forward dosimetry fashion. And for each chemical, you get results. So as long as you chose one of the more complex models, like the PBTK model, you actually will get time course information. So you'll see the concentration of each chemical over time predicted in plasma and each of the tissue compartments. And then you can also look at things like the distribution of Cmax or the distribution of steady state concentration across all of the different tissue compartments that are included there. And then you can also do this in a reverse fashion. So the IVIVE analysis tool, in vitro to in vivo extrapolation, is taking an activity concentration from in vitro assays, again, choosing which types of PBPK models you want to use to do that dosimetry extrapolation, customizing it in the same way. What is the exposure route? What is the length? What is the simulation time? And then extrapolating back from those activity concentrations to give you predicted exposures that would result in internal concentrations that you started with, so where you see activity in your in vitro assays. And one really nice thing about the IVIVE tool is when you get those extrapolated administered doses, so those predicted exposures that would lead to those internal concentrations, you can then overlay actual data from legacy in vivo animal studies or from human exposure biomonitoring data or predictions and compare and look at margins of exposure between predicted extrapolated exposures from in vitro activity concentrations and measured exposures in animals or in humans. So this shows you an example of that output. So you get these equivalent administered dose distributions. So say you put in 20 different in vitro assays, they have different activity concentrations. You use IVIVE to extrapolate back to predicted doses. And then you can say, well, how does that compare with doses from in vivo animal studies? And now you can also overlay exposure predictions from the SEAM3 models to look at chemicals where you may have small or even negative margins of exposure. So just a summary of some of the enhancements that have been added to the latest release of ICE. Again, we added the links to SEBS and the dashboard and the downloads. We have updated the curated high throughput screening data to include OBO Foundry ontology annotations. We have additional data flag information being returned through Curve Surfer. We have several new chemical quick lists. We've improved our REST API, not only to include the OPERA applicability domain information, but also to allow researchers to access the raw concentration response data that is being used to feed into the Curve Surfer tool. And then we have put a lot of time into updating our documentation. That was something that was emphasized at last year's SACADA meeting, is to continue to build out the manuals and the help videos in a way that will improve accessibility for every range of user, from the most naive to the most expert user. So just very briefly, I want to touch on some of our strategic activities that we've undertaken to continue to improve ICE. So we actually hired a team. So we used one of the DCT contracts, actually it was through the NICEUM support contract, through a subcontract to ICS, to engage experts in the toxicology and environmental health science space to do an external review of ICE. And this was nice because these were people who were not already expert users of ICE, but are very familiar with the science behind a lot of the data that's in ICE, and also work themselves with regulatory agencies and many of our stakeholders in the scientific community. So we had some of their folks go in and do a full review of our front end user interface and look at things like functionality, intuitiveness, regulatory utility, things that we should add to our wish list. And then we had, it was actually their VP of software development, an extremely experienced software developer, and his team conduct a full audit of the back end database, schema, and software stack. So this is just a sample slide. I'm not going to go through all of these in detail, but just to say that we got incredibly robust and useful and actionable feedback from this external review that is really helping it, that we're, moving on very quickly and helping us to improve the functionality, the utility, the intuitiveness of ICE. And all of this was presented as part of an all-hands ICE review and planning meeting that we had just last month, just a few weeks ago, where we did a full day planning meeting with the entire NICEDM team and our contractors and the external reviewers that came in and presented to us. And then we had lots of brainstorming sessions amongst the ICE development team and the ICE support team and an ICE user base to really characterize the current state of it, identify challenges and opportunities, and where, how can we prioritize limited resources and really maximize the utility of this tool in the future. And so it was fantastic. I was really happy to see that it wasn't a test of our software developers, but if it had been a test, they would have passed with flying colors. So they came, some vulnerabilities were identified, but in general, they were like, yes, this really fantastic, best programming practices being used, great documentation of the code, using all the right software stacks, really good architecture of the database. So congratulations to Siom for their excellent support that they provide on the back end of ICE, and then the front end of ICE and coding that as well. Excellent functionality. They had lots of suggestions for improvement, but lots of kudos as well. So that was great. So based on a lot of this feedback, based on what we've gotten from SACADM and many of our other stakeholders, we have some items that are very much in progress and ongoing. The EPA just released a new version of the in vitro DB, so we have to update our workflow and our pipeline and our QC pipeline and all of the effort that we do to, curate that data to align with their new in vitro DB release, and that is not a small task. Updating various quick lists, integrating them into our data pipeline. Several features have been requested for the IVIVE and PBPK tools in terms of letting users provide their own data around things like parameters. So users already can provide their own in vitro data as inputs into the IVIVE tool. They can also provide their own in vivo data to overlay and look at those distribution plots, but they would also like to be able to provide, for example, parameter data. So all of this is ongoing, and I'll just stop there and thank the amazing team that provides just incredible support and leadership and direction and application and development of ICE. They're really an outstanding group of people that I'm privileged to work with every single day. And that's not just the people that are pictured on the screen there, but other folks across the Division of Translational Toxicology that we work with very closely, and our partners at all of our ICBAM federal agencies as well. And thanks so much to SACADM for consistently every year providing us with very useful and very actionable feedback that we're trying to take on board to continue to improve ICE. So I'll stop there and ask if there's any clarifying questions.
Thank you. That's amazing. I love it all. Kudos. I had to list of people that congratulate you. Quick question. Does the PBPK have or plans to have or can have an option to select sex even if it is not female or not available?
I thought you might ask that. Unfortunately, not yet, but that is a really important suggestion that, I should say that you don't have the option to select sex, but we do have a population simulator that is incorporated. It's called HTTK POP, and it's incorporated in one or more of the PBPK models that are on the back end of ICE. And you can do things like simulate across a population, and that includes simulations of, distributions of gender, ethnicity, variables like body mass distribution, et cetera. But it's pretty generic in terms, it's trying to really represent the distribution of a population to protect for sensitive subpopulations. But we are very, very interested in better incorporating population variability into our PBPK and IVIVE tools, and the sex as a biological variable is a key part of that. So not yet, but thank you for the suggestion.
So just two quick questions, and I apologize for my naivete up front. So the first question I had was around your stakeholders, and you mentioned method developers, chemical producers, risk assessors. Can you give a little bit more detail on, what specific risk assessors, where are they coming from, or kind of any thoughts on the stakeholders?
Yeah, thank you for that question. So that we have 18 federal agencies that are part of ICVAM, and several of those agencies are regulatory agencies, right? So EPA, FDA, those are kind of the two biggest players in the regulatory space. But then we also have colleagues from, for example, the Consumer Product Safety Commission or ATSDR that do more sort of in that post-market surveillance space and do risk assessment in their own context as well.
Anyone from private industry that would be, I'll give an example later on during the discussion, but.
Sure. So we are increasingly running a lot of ICE trainings at scientific conferences like the Society of Toxicology annual meeting or ASTCT, and more we're getting participation from our industry stakeholders and getting really good feedback on what their needs are as well.
Yeah, and then my last question is just kind of where is the test data coming from? Is this specifically kind of NIH developed, or is this public, private, mixture of everything?
Yeah, so it's all public data. It has to be because it becomes public when it's on ICE. So it's part of sort of the government mandate of transparency and open access. I'm smiling because that was one of the things that came up during our planning meeting a couple of weeks ago was, we have a wish list a mile long, and many of that is around like, well, what types of data should we be incorporating into ICE in the future? What would be the most useful for our stakeholder community? And so we're considering various methods of querying the community for what data they would see as the highest priority to bring into ICE. And I think that's actually one of your discussion questions. Yeah. So maybe put a pin in that.
I will. Thank you.
Thank you.
Okay. I am not seeing any more clarifying questions from SACATM or ICVAM members. So with that, we will move on to the next talk. And it's a pleasure to welcome Dr. Mansouri, who is going to talk about the COMP8 model.
Thanks a lot to the chair, and thanks everyone. Good afternoon, everyone here, and thank you to everyone attending online. So today's talk is about this collaborative modeling project predicting acute inhalation toxicity, which was long awaited for. Thank you. Okay, perfect. This project was long awaited because of all the preparation that needed to be carried on, the data and getting the collaborators together and all that. So this talk would be talking a little bit about the background around the project, a little bit about the data set, and also the modeling strategy. So This is a part of a series of collaborative modeling projects that we started a few years ago. It started a little bit with the EPA actually leading the endocrine disruptor collaborative project for estrogen and androgen receptor. And then we continued that trend with the acute toxicity modeling projects. And we had a lot of success around dermal systemic toxicity and acute oral systemic toxicity, which was modeled as part of the CATMOS project that you probably heard of. And we had a lot of success around that, especially regarding the use of the models themselves and the predictions in the regulatory processes, especially like I mentioned, the estrogen and androgen receptor activity prediction, but also CATMOS. And we just a few weeks ago published a new paper about how that can be used within the EPA and other regulatory agencies to support their regulatory processes. And right now we're moving to the acute inhalation, which is also used by multiple regulatory agencies, not only in the US, but also around the globe. And that's why this is an international collaborative project that involves many entities around the world. So a few years ago, there was this workshop that actually identified how inhalation data is being used within regulatory frameworks and how a modeling project can be brought together in a collaborative way so that all these agencies in the US and around the world can use the existing data and the models later on for their processes. And like you see here, the data itself, even the collection and the curation took a few years, but even after the 2022, we continued working on that data set to make it ready for modeling, which took a little bit of time and a lot of effort from our support contractors. Kudos to them. So a little bit about this type of modeling, which is a bit different from your regular type of QSAR modeling. So this involves many collaborators, different modeling techniques, and then all of that is brought together at the end to build a consensus model, like a ring to govern them all. And that is what is implemented at the end in this suite of QSAR models called OPERA. So the reason we've done this, we carried this type of modeling is, first of all, for the challenges around all these endpoints that we try to model, like the estrogen androgen receptor and acute oral toxicity, and now the inhalation toxicity, which has also a lot of challenges just regarding the endpoint itself and also the data. And that requires a lot of tackling it from different angles to be able to build a final consensus that actually leverages the strengths of the single models and also sort of overcome the limitations of every single model. Obviously, any model you build has certain limitations, certain applicability domain, accuracies within certain classes of chemicals more than others. So that's why we aim for this type of collaboration, and also for the outreach as well, and to maximize the use of the data that we have and curate it. So these are the three main previous collaborative models that we started with, and right now we're carrying the same type of approach for the acute inhalation, although, like I mentioned, it has its own challenges that we try to build the whole modeling strategy around how we can maximize the use of that data set, which is very different actually from the previous projects. Just the overall approach is similar, and also a lot of the collaborators that already participated in the previous projects were also invited to collaborate in this project, but of course we reached out to more. So a little bit about the data set itself. Like I mentioned, our support contractors at Innovative did a lot of work combining data from different sources and doing a lot of curation steps to make sure that it's all the entries and data points are brought to the same standards as our previous projects, and also the type of curation that happens after, or the preparation, I should say, that happens after curating the data itself to make it QSAR ready. That's also another step that was carried on after all the initial steps of curation, of collecting and curating the data set. So I'll not go through all the different steps of curation. And this is actually, as complicated as it might seem, this is a simplified workflow from the different steps that needed to be done to get the data to where it is right now when we started the modeling project a couple months ago. So all the data was collected from the different sources, then the units standardized, the durations as well, the species, and as you can imagine, all the different inconsistencies between the different sources that was also taken into consideration to get the data into a step where it can be modeled and a QSAR modeling procedure can be carried on such data. So the data itself that we ended up with after multiple thousands of entries, we ended up with about 100 entries for about 1,000 chemicals. Most of the data, fortunately, is continuous or LC50 entries for about 780 chemicals, but we also have limit test entries for about 300 chemicals and range entries for about 45 chemicals. So as you can see, the three types of data actually are complementary. So we also use that to further process and curate and make sure of the accuracy of the data that we ended up including in the modeling step. And as you can see, also there is some overlap that also helped us making sure that whatever entries we consider for the modeling are the most accurate of all. And another thing that we also paid attention to is the functional use categories. And I'll not go through all of them, but as you can see, the distributions of the data points among the different use categories of interest, there's a very good and harmonious distribution between all those use categories. And this actually will also be beneficial for a later on or a satellite study to the main modeling project, which is going to be carried out on the variability analysis of the data itself, just like we did with the previous data set on the acute oral toxicity within the CATMOS project. So the modeling strategy itself, it started from, like I mentioned earlier, that workshop that identified the need for inhalation data and predictions and which endpoints need to be brought together in this project, and 5 different endpoints were identified here. The GHS, a couple for EPS, the CPSC, and the DOT. So GHS is the international one, and the remaining ones are within the US. And a lot of them use the LC50 entries for our exposure, but also the different categories and schemas are considered. And the challenge with these multi-categories is that they're not standardized among the different agencies. So you can see there are different categories for each system. And in addition to that, the thresholds themselves of the LC50 values are not the same, as you can see, between GHS, EPA, and CPSC, for example. The other additional thing is that the physical phases, which adds another level of complexity to the project, and especially from a modeling perspective. So, for example, the GHS has 3 different phases: gases, vapors, and dust and mist. While for the EPA, you have gas and vapor together, and then dust and mist as another phase. And they vary also for the with the CPSC and the OPP. So there are a lot of layers of complexity that are specific to this project that we did not face within the previous projects. So bringing all of those together, GHS 3 phases, EPA, OPPT2, the OPP no phases at all, CPSC2 and DOT2. And if we were to consider like a classic way of modeling, we would end up with 11 endpoints if each one of those phases would be modeled separately. However, this is not possible in this case, especially that we were not as lucky as for CATMOS, for example, where we had about 12,000 chemicals. Here we only have a little bit less than 800 chemicals. So with all those complexities and challenges, we opted for a different type of modeling that determines the, based on the phases that each data point is considered later on after making the predictions. Also because we only had about 28% of the data points with reported phases. So that makes it only a third of data that can be modeled if we were to model each one of the phases separately for each one of the endpoints. So the strategy that we opted for is actually to make predictions of the phases of each one of the endpoints, and we carried a little test to make sure that we can make those predictions accurately. And actually, based on the OPERA predictions for PhysChem properties, we were able to very accurately predict the different phases of the data, the 28% or the third of the data that we had reported phases for. And that actually solved the whole problem of making or building the models, and that simplifies it a lot for us. So we ended up deciding to model the LC50 point estimates in 2 different units, milligram per liter and ppm, and that helps simplify the modeling steps, and I'll show how the whole strategy will be put together after this slide. So in this step, we divided the data into sets, so a training set that represents 80% of the data, about 600 chemicals, and then 20% of the data as an evaluation set, about 150 chemicals that were left for later on validation of the different models. So the training set was already delivered to the modeling groups, the participants, and the evaluation set was held out. But at the same time, you also delivered the participants a prediction set, which is a list of chemicals that we want predicted that represents a list of interest to different regulatory agencies, and that list that we have interest in making predictions for. And also this is like we did for CATMOS, and this actually, most of it is a legacy from CATMOS, is to help with the evaluation and validation of the different models later on. And the evaluation set is actually injected in that list, so that later on we have the predictions of the evaluation set without having to rerun the models. Finally, the proposed modeling strategy turned out to be really simple, which is providing the data sets to the modeling groups, asking them to model it within using the LC50 values for 2 different units, and then combine all of those into a consensus model and apply a weight of evidence approach for consistency, and then use the PhysChem properties that are predicted from OPERA and apply that to determine the physical states of each one of the predictions, and then based on that determine the classes or the categories for each one of the regulatory systems. And all that would be implemented into a unique consensus model in OPERA, just like CATMOS. So the only thing you would need to make the prediction is actually the chemical structure itself, and then the model will be a consensus hybrid model that will actually, in the background, run the predictions of the PhysChem predictions on that same chemical and determine the LC50 and then assign it to a specific category within each one of those different multi-category systems. We're expecting right now the different groups to deliver the models and the predictions. And later on, we will conduct an evaluation criteria, which we already defined its steps that are based on the 5 OECD principles for QSAR validation. So the defined endpoint, a simple algorithm, so as simple as possible to make or to help us with building the consensus model. A defined applicability domain, which is very important because we only consider the predictions within the applicability domain. Appropriate measures of goodness of fit. So we apply 2 different levels. So the first level is based on the single model, and then a second level will be to validate the consensus, and then add the mechanistic interpretation if possible. So we know different groups have different approaches, and we understand certain are more interpretable than others, but the final consensus will be implemented in a simplified way to provide some mechanistic interpretation about how those predictions are made and how do those relate to the most relevant analogs from the data itself. So applying the endpoint-specific similarity to make sure that the chemicals that we provide as nearest neighbors for comparison are actually relevant to this specific endpoint and not just general similarity. So like I mentioned, the timeline, and this is a very optimistic timeline. by October 1st, we will have the predictions and first summary descriptions of the different models. And then after that, we will conduct the evaluations and combine the models into a consensus. And just like we did with CATMOS, Syrup, Compara before, we will write a manuscript describing the whole process of this project, as well as the implementation of the model within Opera and how to use it to apply it in real predictions. So the participating groups in this project were about 20, and I want to thank all of them for their efforts so far. And we still have a lot to do, but I'm confident that we're going to get there. Hopefully in 2025, we have a model that is implemented and a manuscript that goes with it. And with that, I'd like to thank you all and thank our team and all our collaborators. And I'll be happy to take any clarifying questions.
Okay. I have a clarifying question and others, please use the raise hand feature and you can go next. You had put substances on the market from EPA. What can you tell me what that is?
Oh, that's a list of chemicals that the EPA identified and put on their Comtox dashboard, and includes all the list of chemicals on the market. I think it's not just in the US, but around the world. Yeah, it includes the TASCA, plus it's more than the TASCA. So includes all TASCA and other chemicals as well.
Great, thank you. Dr. Benham?
Yeah, I have two clarifying questions. The first one is that the LC50 or LD50, you mean the lethal dose, 50%, am I correct? The endpoint that you
concentration here.
So in the cellular biology, the cell death is kind of the ultimate impact of the injury. Are you planning to do for milder endpoints, cellular stress or other sort of like biological response, before we reach the cells reach to the lethality?
We don't have that data. We don't have that level of data. No. Right?
Can I also clarify? So the LC50, it is a regulatory guideline test that's part of the acute toxicity 6-pack of tests. So there's 3 acute topical toxicity tests and then 3 acute lethality tests. And it's the LD50 by oral or dermal application, and then the LC50 by inhalation application. So it's a very specific regulatory guideline test that we have compiled, lots of data from, that we're using to create a model that could potentially replace that specific test guideline. To your point about other endpoints in inhalation toxicology, respiratory toxicology, that's certainly of interest, but that's not in the scope of this particular project.
Got it, understood. And the other clarifying question I have is that when you have your models in your training, do you include negative controls, or you always look for just positive controls or compounds that they will have some degree of cytotoxicity?
Yeah. Go ahead, Emily.
So the data that we received were from negative tests as well as positive tests. So we had, as Mal explained, we had some upper limit tests. So no lethality was seen at the limit of the regulatory test. So I think, 5,000 mg per cubic meter as one of your concentrations. And so, yeah, there's negative data in the modeling set as well as lethal data. The one thing that we are missing from the distribution is we don't have highly acutely lethal data. So it is skewed a little bit to the moderately to low lethality. And so that is one of the shortfalls with the data set, is we just couldn't get that really, really acutely toxic data because it's just not out there.
Yeah, thanks.
I forgot to introduce myself, Emily Ranke, Innovative.
Thank you, Emily. I'm just going to remind everyone that it is clarifying questions only at this time, and that we will be holding all of the discussion until the end of the session. But Dr. Miles, would you like to give your clarifying question?
Yes, thank you. So very nice presentation, Dr. Mansouri. I did have a question around fragrances. There was a category name there. Can you speak a little bit more about how substances are put into that category? Because we know fragrances can be very complex mixtures with hundreds of substances. So if you could speak to that.
This is Emily Ranke with Innovative again. There are no mixtures within that data set. It's all single chemicals.
So even the fragrance?
So I think there are probably there's some fragrance ingredients that are single constituent ingredients, but there are no mixtures within that data set.
Okay, thank you.
Dr. Ellison?
Yes, so thanks, great presentation. Can you just clarify if it was more than one species that were going into the data set? And then if it was multiple species, was there any need to adjust for inhalation rate?
Hey, once again, this is Emily Ranke with Innovative. So the only data that are going into this modeling project are from the acute rat test. We did collect other data, but that was cleaned up as part of that, cleanup and curation process. So we tried to keep it to just the rat that would have been used for the regulatory guidelines.
Okay, great. Thank you.
I was going to say to answer the other part of your question is we did standardize everything to that 4-hour exposure as well. So we had some shorter and longer exposures and adjusted by Haber's law for that.
Great, thank you.
Great. I am not seeing any additional clarifying questions at this time. So with that, we will hear again from Dr. Klaus-Troyer, and she will present on camera.
Okay, great. Thank you so much. Someone is advancing my slides for me. All right, so camera, you've heard me, spoiler alert, allude to it a little bit over the last 2 days, but I'm really excited to talk to you about it now. It is a new initiative, a new resource that we are launching. It's called the Collection of Alternative Methods for Regulatory Application, or CAMERA. It is intended to be a new interactive database of validated and qualified NAMs. And that is not an official graphic associated with it, but it is a graphic that I made using AI when I was playing around with all the different concepts that I would like to be included in CAMERA. So I just thought it was a nice way to start the talk. So this was proposed and actually requested by our exam federal agency partners, and I think more broadly across the scientific community. We certainly heard this as a strong recommendation from the ACD NAMS working group. So the presentation that Ellen gave us yesterday highlighted several recommendations around the need to facilitate access to validated NAMs and the information associated with their validation and their acceptance or their consideration by regulatory agencies. And so we had a discussion at across several exam teleconferences to see whether or not there were existing resources that could be sort of repurposed for this, or that we could partner with others. And what really came out of it is a real dedicated need, an urgent need for a dedicated resource that would be kind of a one-stop shop where people could go and access information on NAMs, for specific endpoints that would be considered or accepted by specific agencies for specific chemical categories, et cetera. And so what I did is I went to NIEHS leadership, and they have an internal funding process with year-end money where they consider various proposals for funding, and they have very generously agreed to fund the initial beta version, the development of this app. So it will be a front-end web-based user interface and a back-end database that will include validation data. And I'm going to get into much more of the details around what we are envisioning, but really these slides are a list of questions to all of you in terms of what you think would be useful, both on the front end and on the back end. Also, very graciously, DTT and Heather has agreed to support a contractor position as a senior project manager. We actually have a really, really excellent person who has accepted that role. I just found out yesterday. So I'm really excited about that. And then the plan is that NICEDM will, so once we have that initial influx of resources to stand up the first version of the web-based interface and the database, then NICEDM will continue to sustain out-year maintenance via the support contract. Obviously, we have a portfolio of computational tools that we want to ensure interoperability with. So ICE is obviously one of those, but you heard yesterday about complementary. So the data hub that is envisioned under complementary is going to be a place where all of the initial data from the NAMs that are funded under the complementary tech development centers will reside. It'll be, a variety of many, many different data types and from many different types of methods. Not all of those methods will enter the VQN, the Validation Qualification Network pipeline, but those that demonstrate real added value and robustness and are technically characterized will go through validation and qualification. And once they go through validation studies, then the idea is that they will land in camera. So camera will be sort of the landing place and the home for NAMs that come out of the complementary program and hopefully other programs that will be funded by other federal agencies as well. And then, of course, we want to ensure interoperability with some of our other federal data resources like SEBS and then others. I would welcome feedback on that too. So, as I promised, these slides are a series of questions. So first, thinking about the content of Camera, what should be included in that back-end database? What should users be able to access? So a couple of ideas here, the types of different NAMs that are available, what stage of validation those NAMs are at, the actual validation study data. To me, this is one of the most important elements that will set Camera apart from other resources. And I'm going to highlight a resource from our partners in the EU on the next slide that I think is complementary, but very distinct from what we're trying to do with Camera, because I would really like to, and it's going to take a lot of data curation, but I would really like to give users access to the actual data that went into validating those NAMs, so that they can do their own analyses and potentially be able to see whether or not the chemicals that they're interested in testing in those NAMs, fall into the applicability domain and do that in a data-driven way. Acceptance or guidance, around which agencies or which centers or which offices within those agencies might consider or accept those NAMs for various use cases. Protocols for those NAMs. Should we have an actual repository of protocols that people can download, or should we have links out to external sites where those protocols are maintained by the method developers? If we have the protocols ourselves, we're going to need to version control those. We're going to need to, be able to provide information on their level of acceptance and when they were approved and what version that is. And it's going to be a lot of work to keep them updated. So obviously I have a bias of what we should do around that, but it's an open question, right? Information on the applicability domain of the NAM, context of use, protocol variations, et cetera. We did consider under the ICMAM committee discussions, whether or not the existing TSAR, which is the tracking system for alternative methods toward regulatory acceptance that is maintained by our friends in the EU, Ural ICFAM, would meet our needs, and decided that it would not. First of all, there was a strong preference on the side of our federal regulatory agency partners that this should be a system that is maintained by the U.S. government and is applicable to U.S. regulatory applications first and foremost, while maintaining international coordination and harmonization. But also, while TSAR is an amazing resource, it is not a database. It is essentially a list of assays and where they are at their stage of validation or acceptance. And it has lots of useful information, but it doesn't have the functionality and the sort of interactive user interface that we are envisioning for camera. So there was, a clear, dedicated, unmet need here. So what should that functionality be? So how should users be able to interact with camera? How would you want to be able to search this database of NAMs? I assume that people want to be able to search by endpoint, so show me all of the NAMs that I can use for eye irritation and corrosion, or that I can use to screen compounds for developmental toxicity potential or whatnot. Potentially by agency. So show me all the NAMs that FDA will accept for skin sensitization for cosmetics, for example, or will consider. By availability, that's tricky, right? As the government, we can't necessarily, we can't provide links out to CROs or certain assay providers, but at least having some level of information on sort of regions in the world where these platforms are available, I think would be useful. By R, and the reason I put that up is because at the moment we just have a list, a static list on our NICEDM website of alternative methods, and one of the pieces of information that you get there is whether or not it's a reduction, a refinement, or a replacement method. And so that might be something that users might be interested in being able to search by. One of the ideas that I had that I'm particularly excited about, I don't think we'll be able to achieve it in the beta version, but maybe in a subsequent version, is along the lines of ensuring connectivity with ICE and all of the amazing work that Kamel leads under our computational chemistry program, is leverage these cheminformatics tools that he and his team have developed to actually provide workflows where people can have a list of chemicals that they're interested in testing in various NAMs, and use those cheminformatics tools to actually look at the similarities of the chemicals they want to test with the validation study compounds that were used to establish confidence in the assay for a particular context of use, or any applicability, domain constraints, things like physicochemical properties. There may be ranges of solubility that are required for a chemical to be tested in a NAM. So have those tools be part of the camera interface in a way that leverages all of the cheminformatics work that we've done. And then, of course, what should that interface, what should it look like? We want it to be intuitive. We want it to be user-friendly. How much of an emphasis should we put on just straight up data presentation, downloading flat files of validation study data versus trying to invest in programming graphics? That takes some heavy lifting sometimes. Would it be useful to have kind of a report functionality where users could actually download a report that incorporates the validation study data, some of the comparisons they've done with their chemical lists, et cetera, that then they could take to as part of submissions to regulatory agencies? And what would agencies want to see in the content of a report like that? So these are all just ideas and, just to stimulate discussion. Process. How do we select and prioritize NAMs that should be added into camera, into the database? What should the criteria be? I think at a minimum, these need to be NAMs that are at least technically characterized and have gone through independent review. Do they need to be fully validated and accepted by one or more regulatory authorities around the world? Or is it acceptable for them to have some degree of technical characterization and validation within a particular context of use? Obviously, we want to see alignment with the concepts in the exam guidance on validation, qualification, and regulatory acceptance of NAMs. The good news is that we are, it's not like we're starting from scratch. We have a backlog of over 100 methods that are already listed on the NICEDM website as accepted alternative methods. Many of those are OECD test guidelines, and we've already undertaken, or at least started to undertake, a curation effort in association with a related project where we're actually looking at numbers of chemicals and what types of chemicals have historically been used to validate OECD test guideline methods, both in vitro and in vivo. And so the innovative Nicedum team has already started identifying and extracting a lot of the data in those in vitro validation studies for the OECD test guidelines. So we have a really good springboard in terms of data that we can put into that back end. One question that I have is, out of the 3 Rs, many of the methods that are listed on the Nicedum website are refinement or reduction approaches. Should we, I think those are important, but should we prioritize putting replacement approaches, so non-animal NAMs, as sort of our first priority to put into this database? Again, open question. Beyond OECD test guidelines and those that are already listed on the Nicedum website, where else should we go to identify NAMs? I meant to put complementary on here, but complementary is intended to be a pipeline of NAMs that will come through the VQN and will ultimately land in camera. But other sources might be ASTM, ISO, US Pharmacopoeia, our ICADM partners, not just ECVAM, but across our international cooperation. We've also thought about putting out not a survey, just asking our agency partners and other stakeholders for prioritized endpoints. And we also wondered whether or not it would be helpful to have a user-based submission process, and who would we accept submissions from, and how would we review those submissions? Do we inherently, under the international cooperation of ICADM, do we accept validation of methods that has been undertaken by our ICADM partners? So mutual acceptance of validation, similar to mutual acceptance of data under the OECD. So those are all some thoughts. We also have to think about governance. I think we have answered this first question, because we already asked ICVAM this first question, whether or not we should have an ICVAM work group that had a defined scope and charge and then inherently has a finite timeline of working towards fulfilling that scope and charge and then gets sunsetted. I think we've decided that that's not the way to go. We intend for this to be a resource kind of in perpetuity that will be maintained in out years. And so we are in the process currently of forming a steering committee that will be a standing group that will help inform on the development and the process around camera. That is likely going to be fed only, but we also have thought about having a workshop to solicit input from our stakeholders across the scientific community to ensure that this is a broadly applicable and useful resource. And then, of course, governance is linked back to process in terms of how often will we sort of revisit. Is it just an ongoing rolling process where we're looking at including new NAMs, or are we going to kind of have an iterative, every 6 months we do a landscape assessment of what's out there and what needs to be brought in? So I will stop there and ask for clarifying questions.
So exciting. Okay, I'm selfish, and I have some clarification questions. I have two. For the first one, I don't know if this is to Unical or one of the other ICVAM members that had input, but do you anticipate that this could be a tool for regulatory agencies? For example, I'm going to call out EPA, that can point to, instead of having to publish separate guidance, because we know that from a resourcing perspective, sometimes that's difficult, or would this be in addition to that?
A great idea, Catherine. Thank you. I can't speak for EPA. I can't speak for whether or not they would feel comfortable, pointing users to camera and saying, hey, if it's in camera, then we'll consider it. I think that's going to depend on the office and the sort of legal jurisdiction that they fall under. But I hope that it will be a resource in that capacity. Rick.
Hi, this is Monique Kern, CPA, as Nicole pointed out, obviously it will depend on the program and their needs. I know from our perspective in the pesticide program, anytime that we can build off of what's already been put together, we would like to do that. Although at the same time, I know that there's often confusion or support, whichever way you want to go with it, for us to clearly state all the time when we are willing or not willing. The difficulty there is obviously a resource constraint, and the hurdles is always putting out those documents over and over again. I think in our program, we're very lucky. Our statutes are flexible and allow us to consider, a range of different types of data. But it is always going to be a challenge if our stakeholders are still hesitant to utilize it. So we often try to communicate that we want registrants to come to us, if they have ideas, especially, thinking outside the box, great, come to us, let's talk about it. Early engagement is always really helpful, especially when some of these things are still in development, but we still think that they could be useful along with other information. So it really is dependent on the case and the program and all of the fun things that I get to say are nuances, but I do encourage if there is any kind of resource, whether it's an in silico model or other, that they engage with us early, even if it's not been used before. We have lots of examples of that, and we've collaborated quite often across different agencies, particularly with NICEDM on different projects.
Thank you. I know it's not just that it's a similar situation for FDA too, and I'm sure others. So thank you for that. My second question is, what would be the process for getting new data into camera? Like, who would nominate that, or how would you?
I believe I had that same question in my slide. I think it's an open question and something that we need to work with our federal partners on. But it's something that I would love feedback from the committee on what your recommendations and what your perspective would be on how we should establish a process. And I think we have to have a process. I think it has to be very clearly articulated. But I think it's still an open question what that will look like.
Great. And Rick, did you have a clarifying question or something that you wanted to add on to?
Just a quick clarifying question for Nicole. So I'm still trying to get my head around TSAR versus camera and potentially other databases that are out there. One of the greatest apprehensions that I have is that different programs doing very similar things then come up with databases of their own. And then the global biomedical community is left with a heterogeneous collection of different data repositories with some stuff in one, some stuff in another, without a lot of seamless integration. So what's the state of that? Is TSAR, is camera sufficiently flexible in its developmental stages that it could accommodate some of the needs of TSAR, and that camera then becomes more of a central repository for the global community to deposit the data?
Yes, absolutely. And so that was one of the things apologies that I didn't highlight it in a detailed enough way, but we did a deep dive into TSAR. We had our ECBAM colleagues present TSAR and their development and maintenance process at our ECBAM teleconferences, and really concluded that there are really dramatic differences between what we're envisioning for camera, particularly the database functionality and the ability to search by things like endpoint or chemical similarity or agency or program that might accept the method, none of which you can do in TSAR right now. That being said, we are fully engaged in harmonization with ECBAM and our other ICADM partners to ensure that methods that have reached the stage of validation where they would be eligible for and applicable to, inclusion in camera, we'll work with our ICADM partners to get the data in shape, and put it into the camera database and have linkages back and forth between TSAR and camera. So they really serve very different functions, but they will absolutely be connected and coordinated.
Okay, sounds good. Yeah, I did hear those details. That's part of the reason why I asked my question. But it's good in the end, making sure that all these data repositories kind of seamlessly connect together, because having heterogeneous sources of information where investigators may not know where to go, and so I think it's just something to pay a lot of attention to at this point.
Absolutely, thank you.
Sally, go ahead.
Yeah, so I just clarifying question. I think I heard you say that primarily what's available in a current database is reduction and refinement. And then I'm also just kind of play on words. Would those be considered VAMs, validated alternative methodologies, that might be more palatable when we're talking to our regulatory people? So is that what you said that what's currently available not in camera, but in a database?
So right now there is not a database yet. There is a list. There's lists on our website. There's lists in TSAR. The list of accepted alternative methods by U.S. regulatory agencies that exists on the NICEDM website. Again, no database associated with it, just a list of the methods themselves, which agencies accept them, and whether they are a replacement, reduction, or refinement approach. I just noted that quite a few of those are refinement and reduction approaches, so variations on the, acute oral LD50 using a different procedure that, decreases the number of animals that needs to be used, et cetera. Refinements to the LLNA to decrease pain and distress for the animals, things like that. And so the question was, we have to have a prioritized order of NAMs, validated NAMs that we're going to put, or VAMs, but we like to refer to ourselves as VAMs, ICVAM, ECVAM, COCVAM. So that might get a little confusing for a different way. But anyway, the validated NAMs, there has to be sort of a hierarchy of what we do first, because we have to put our resources, in where we're going to sort of get the most bang for our buck. And so my question was whether or not we should be prioritizing the replacement NAMs or the non-animal NAMs, the in vitro, the in silico, the integrated, the defined approaches, for the first tier of putting into camera, and then consider refinement and reduction NAMs as sort of a second tier, or not, whether, we have to have a prioritization process to work through the list. And so what should that process be based on?
This is Heather Padisall. Sorry, my Zoom crashed, so I physically raised my hand in the room. So we heard earlier about the DNT battery, and then FDA is trying to take it up, but they've made some changes. It's not exactly the same. So how would that be documented, and how would something like camera handle that? Because there's going to be drift in these protocols over time, partly out of necessity and practicality. But have you given some thought about that?
Yeah, absolutely. I think it's a really important point, and that's why I sort of alluded to the fact that I would really like to work with external groups, and rather than having a static version of, time-stamped protocol that people download, I would rather have a link out to a site that the method developers themselves, or the people that are applying the method or are, updating the protocol, they would be responsible for maintaining it and ensuring that the version that people access through camera is the most current and up-to-date version.
Thank you for that clarification.
Dr. Barnes, do you have a clarifying question?
Just more of a comment, Nicole, to your question about prioritizing. I mean, one of the way to prioritize potentially because you're looking to have the access to the data, right, being shared. So it's maybe that's where it's going to be prioritized, right? Who is willing to share that data for those specific tools versus, which one of the 3 Rs would be there. And yeah, so that might be one of the ways to help you make those decisions potentially.
Great, thank you. So we're going to move on to the public comments section. So all public comments were solicited through the Federal Register notice, and written comments were posted to the meeting webpage and distributed to the Setata members in advance of the meeting. Individuals requesting to make oral comments were required to register in advance for planning purposes. I would like to indicate that one written comment was received for this session, and that was from the Humane Society of the United States, and there were no requests to present oral comments for this section. Okay, and with that, we will move on to the discussion section for this final session of the day, and we will start with the lead discussants to provide their comments on the computational resources. I don't know if you guys decided what you want to go first, but okay.
You mind if I go first?
Adrian has put his hand up.
I just want to go while I have these kind of fresh in my mind. So first of all, I think, that's kind of been the theme for these last 2 days is to see just so much accomplished over the last year for this week. It's pretty impressive. And I think there's still work to do for a lot of things, and I'll kind of touch on some of the things. I think one of the big things that I'll have in my comments is kind of the idea of thinking outside the box for end users, not just the government regulatory agencies, and I'll speak a little bit more specific. So going to the first question about functionalities and data sets with ICE and kind of suggestions, I think, for me, the first company I brought up in the clarifying question kind of session is thinking a little bit different about who would be risk assessors that could use something like this. So I think for this, I would open it up to anybody who's doing risk assessment in either within the government or within private industry. And I'll give a couple of examples of how I see that kind of sorting out. I haven't had any time to kind of really tinker with it, but just from what I could see, it could be kind of a full-time job to learn how to use something like this, to do it the right way, to really impact whatever you're trying to do, whatever specific risk assessment. So my question, and you answered it, was, are there any kind of learning opportunities, training, online courses? And you mentioned that some of this was going on at conferences. That's a spectacular idea. So anything that you could do to make that a little bit more user-friendly, I think would be useful. I don't thoughts about, kind of trying to keep ahead with technology and being able to incorporate chat GPT type kind of functionality to something like this would be useful, might help a little bit with the user interface. Because the way that I see this happening, at least from the private industry kind of side, is let's say you have somebody developing a molecule for whatever purpose, pharma, product, whatever, and they have done their big high throughput screening. They have families of 4 to 5 kind of similar chemical structures. Would they be able to put those kind of into this system and, ask it different queries, which of these are endocrine disruptors, and kind of try to get some data to be able to help prioritize where they would focus their additional chemistry to. So, one of the things I think it'd be useful to be able to do this kind of in high throughput and put lots of compounds into this to check for similarities. And then I think the other things that I would kind of think about are more kind of unicorn type ideas of, at a lot of these companies, they have their own internal databases that are very similar to something like this as they do a lot of their medchem. So it'd be interesting to see if they're willing to kind of share how they store a lot of the different things. And then maybe even take an approach of create a large library of compounds with, very distinct chemical structures to help kind of inform and build up the database, things that, a lot of these companies kind of already have, but just trying to figure out a similar type approach. And then my last kind of comment is just, how applicable is this to, let's say, another, somebody wants to come in and use this framework to be able to put their own data into it. That's something that, is amenable. So with that, I'll stop and see if there's any questions or on that before I move into the next set of. All right. So my next set of questions is really on the last presentation. And again, just amazing to see something that when I first started in industry in 2008, I thought, well, this be nice if it existed as a young toxicologist trying to figure out what assays are available to me, what to do, and different kinds of things. So to see this kind of actually come to fruition was kind of impressive. I guess, the first kind of question is, this whole idea of already the 130 that you had mentioned kind of already listed on there. I got a chance to kind of tinker around with that site this morning. It's the first time I'd ever done it. I found it very useful, very easy to kind of do it, kind of from the first approach. And I just kind of went through and looked for things that I had, been interested in the last couple of years, like CAR-C or developmental tox, and was able to kind of quickly get a really quick update on kind of where that is. So I thought that was really amazing. I think one of the things that I would add to it is the functionality to be able to kind of search based off of the regulatory agency. So all of the FDA kind of approved or discussed about assays in one bunch would be useful. I'm not sure if that's already into the system, but I couldn't figure it out quickly. But I think that would be useful because if you're someone that's doing product safety, you probably care less about, a certain set of assays that are very pharma specific from there. And then I think the other piece in looking at that is, I know from my own personal experience that there is a lot of assays that industry use to help kind of make decisions, thinking in the cardio space with isolated rabbit heart, anesthetized dog, cytokine panels, kinome screening, all these things that are typically done, not just across one company, but multiple companies that could kind of be in that as well. So I think, it's kind of the big theme of where would all of these kind of ideas and assays kind of come from. A lot of this has already been published by these companies. And not only do they use things like mitochondrial tox assays, but they published on them. So, I'm assuming it's kind of free for all at that point. And then I think, the next piece, again, whatever you can do to kind of incorporate any kind of AI type interface is going to be useful. I think the piece on fully validated versus technically useful is an interesting question. And I think, there are things, at least that I've dealt with in my career, that may not be fully validated, but were definitely very technically useful in decision making. And after people got comfortable with them, they then became fully validated into something used like hurt binding, or is a good example. Ames assay for gene tox. And then I think the comment that you made on refinement, reduction versus replacement is very interesting. I've never really thought about it in that way. Should we focus on one or the other? I just kind of always pull them together in the way that I thought about it. And the way that I look at it is that replacement is probably what is the holy grail, where we want to get at some point. Most efficient, least use of animals, and if it can be the best predictor, great. But I think refinement and reduction could be the road to get to replacement. So I wouldn't quite roll all of that out because at some point we need to get good enough where we can do that and get to replacement. So that's the way that I would just personally think about it. And I think, trying to figure out what other things could go in there. Obviously, I've kind of restated this many times. I would try to think outside of the box and look to industry to see kind of what things they find useful in their course of business. And the way that is typically thought, at least from my perspective, is that it's often regulatory down. So we approve of this assay, and everyone kind of below says, okay, this is what we can use. Kind of thinking of it the different way, it might be more useful to think about it is, what is everybody else doing that could be considered to kind of push up to get somebody to think of a different way and a different kind of approach. So I think that's something kind of a different way of thinking, but there's a lot of work being done at the end user stage that's being used to make big critical decisions. Has a lot of data behind it, a lot of multi-company use that could kind of be filtered on the way up. The workshop, I think, is a great idea. Kind of get to my last 2 points here. And, all I ask is kind of think outside of the box of who you would bring into this, not just regulatory governments, but regulatory agencies, but all the different kinds of things. And I think, to Catherine's kind of point, trying to figure out how to convince an agency or regulatory agency on something, it might be difficult for something like this, but I think something like this that's well curated, lots of diverse opinions could be a gentle push into that direction to get things started. You kind of have to have everything organized first in a nice, useful way that lots of people can use across multiple industries before you can kind of start taking that case. So thanks a lot. Appreciate it, and great work.
Okay. Hi. So great, great session. So my comments will kind of be all over the place. But to address ICE, I'm definitely not a toxicologist, so I don't use it. But I've, over the past 4 years, watching how much progress has been made has been really exciting. And especially with the uptake, that you can track that. I think it's just been really interesting to kind of see the progress. And how useful it is, and how you're, actively updating it and actively, keeping it current. I think it's amazing. Just a suggestion. I don't know if you have to put the data set in there for cardiotox, but that could be an interesting one, especially if you start going down the, highlighting that. With inhalation data, I thought that was just an amazing, a huge effort, and a really great example of how hard it is to really combine data sets, and that it's not just we're gonna upload it and it'll be fine and we can start modeling tomorrow. So that's also been just a huge amount of work and greatly appreciated. I'll focus most of my comments on camera. I think that's where I can add the most value here. First of all, I agree. I love this. This is great. We need it, and there's critical mass now. If I take a big step back, one of the themes, actually very similar, that I've been thinking about a lot over the past 2 days is the end user. And I think with all of the amazing work and success and sort of this huge effort, we're at this point where you can really start to think about the end user and what does that look like, because it just seems too kind of far out there to worry about it too much. And when I think about it kind of coming from industry or investing or method developers in the private sector, there's sort of these 2 or 4 work streams. They're sort of kind of what was saying around, it's sort of the top down. It's sort of what you were doing, which is let's see what we can do to get regulatory acceptance. Because once regulatory is acceptance and maybe even ask for it, it'll start to trickle down into industry, and the users will use it because of the regulatory decision. So there's that way. And then there's kind of I guess we call the bottoms up, which is in parallel. Method developers in little companies or in academia or whatever are interfacing with pharma or other biotech companies. And again, I'm coming at this more from pharma. And we're working with the scientists, and they're asking for studies, and they're running compounds, and we're generating data. And there's no regulatory bar, so they use them. But they're all doing their own validation over and over and over again. And it's really time consuming, and it's really frustrating. But it's you get paid and you generate revenue, and that's good. And so there's that happening at the same time. And so there's that piece, and then the other dimension is sort of kind of more chemical EPA leaning versus sort of drug FDA leaning. And so I feel here we've kind of leaned towards kind of EPA more, for I think obvious reasons. And I thought what was interesting yesterday was with the Common Fund project and the Foundation project, all of a sudden there's going to be a bunch of pharma type convenings like this that'll be very complementary. And so anyway, it's a long-winded way to get to what I think about maybe what to consider for camera. Personally, I kind of like the idea of just start with it's a non-animal test. I just jump over it, but again, I'm kind of biased that way. So some of the obvious things we already talked about, organ endpoint, cell type, that the sourcing and is it commercially available would actually be a good one, because you get stuff really quick. The other thing that will start to happen is the complexity question will become really real. Is it 2-dimensional? Is it 3-dimensional? It's for the same endpoint. You can have a wide array of different models, and that complexity also kind of maps to throughput and cost. So the higher complexity kind of usually lowers this, but usually higher cost, by a lot. So there's this trade-off between, and so there's a continuum. At Tara, no one ever came to us first. It was let's do 2D cardiac. You're not gonna be the first call. But at the end of the day, there was data that only we could generate. so there's that kind of, it's kind of nuanced, but I think it could be like it's an interesting variable. The other thing that will happen, I think, as you bring in more of like the FDA side is healthy models versus disease models. I think that'll be a big topic. And that it also goes to the market. So is it a tox market or is it a discovery efficacy market? It's two different groups. It's a whole different, scientific community per therapeutic area. And so that will start to kind of, I think, emerge. Support for human relevance. So, to your point around like whether it's papers or whether it's support, but what is it that makes you human relevant? If depending which direction. I would say, another orientation is sort of like a chemical point of view versus a biology point of view, and sort of how to marry those and need both. And then, because some products won't be chemicals, they'll be biologics, they'll be antibodies, they'll be non-chemicals. And so it's just that modality piece will come into play. It's something to think about. And then the level of validation, level of regulatory acceptance, et cetera. I also had kind of commercial readiness. Is it in academia? Is it in the government? Is it commercial? Does this entity have customers? What kind of customers? Like how accepted is it? Is utility use and is a kind of a good measure? No one's going to pay for this if they don't need it. Like pharma or large companies, if they can do it themselves, they're gonna do it themselves. So that was just some like a laundry list of things to think about, and I think the approach around like I think these other efforts you'll start to really get a lot of like the inputs, but working with like the 3Rs organization or like the MPS, working groups and stuff, I think you'll pull in a lot of other technology. If you don't mind, I think I was going to say some stuff yesterday at the validation. We ran out, so I'm going to touch on a couple of things that are kind of related, but a little bit of a divergence. But I think, it's related to this. I think as you think forward and planning ahead, sort of like what does success look like? And I think this idea of like user uptake is a measure of success or a metric and we're doing all this work. And if at the end of the day, no one uses them, that feels like not great. And I think that the challenge is like in industry, it's going to be a cost, time, risk first before it gets to anything about animals. And so I think someone mentioned it yesterday around like return on investment and whatever that looks like. But that I just think knowing that and getting that point of view, I think will be helpful. That said, I do think animal awareness is at an all-time high, and I do think corporate America is conscious of it, and it is something that I think in the past you couldn't leverage very much, but I think like you could start to do that. So I think yesterday someone mentioned something about communication or messaging and the parts of the messaging and communications and how to sort of tap into, I think to your point, the generation that really is more sensitive about this issue than maybe like my generation or like older people. But that's, so I do think there's an interesting sort of communications piece that ties into adoption and users and all this. I think that's really it. And then I would just like to say thank you so much. This is my last meeting, and it's been a pleasure. And I have to put just one anecdote. It's my first meeting. We talked about bridging to the animal data sets. And I remember just being like, this is wrong. Like, we can't, that's not the goal. Like, they're animals and it's highly variable. Like, that's not it. It's like, we should be thinking about what's human relevant, what's physiological relevant. Like, that's, we're trying to like model humans. And so it's been really nice to see, like, over the years, like, that narrative really shift. And I think it's really important. So.
Thank you so much. Just to process, we will get some time, if everyone can stay to the end, to have some closing remarks by the Sakata members. So you will get a chance to mention something at the end. I'm going to pass it over to Dr. Allison for as a lead discussant.
Great, thanks. So this being my first SAC-ADAM meeting, I'm happy to hear all the presentations. Sorry, I couldn't be there in person. but I'll try to make my comments concise here. So very impressive, all the different computational tools. I'm really excited to hear that this is a standing agenda item because I think it's that important that it should be. Just jumping right into ICE, I think great progress there. I like the tool, I like the functionality. I think there's a couple places that potentially things can be either added or extended. So one of them, and I'll just run through them quickly here. So one of them being the chemical quest tool, which uses fingerprints and then Tanimoto similarity score to determine similarity. My company's done a lot of this, and I think the scientific community's done a lot of this, of looking at the robustness of just looking at chemical similarity with Tanimoto score, and oftentimes it being insufficient because it doesn't take into account biological features, and then it's often yielded poor results. But we get better results for identifying chemical similarity when we look at chemical structure, along with metabolism pathways or rates, reactivity, and phys-chem properties. So when you pull the different features together, you get much better success. And I would think that ICE is in a perfect spot to do this because all that information is pretty much housed within ICE. So I think there's an opportunity to extend that chemical similarity feature beyond what it is today. Another possible extension is within some of the in silico alerts. So it was good to see that. And I think the QSARs are nice. It'd be also nice to try to integrate some expert knowledge within that. So there's some decision trees that are out there for different endpoints or for organ toxicities that really have leveraged a lot of expert knowledge to then make decision trees. And those then being amendable to automated processes. I think having that within ICE would be a nice feature as for some of the alerts. I had as a comment about chemical mapping, and then Nicole nicely went over the chemical characterization module of ICE. So it's good to see that. I think the chemical mapping and spatial mapping is very important. Also excited to hear some of the new features regarding being able to input user-specific parameters, especially around the ADME and PK side. As you do that, you may also want to give consideration to allowing for saturable metabolism within the PBPK model structure. So I believe a lot of it right now is just linear intrinsic clearance. We know that a lot of times the tipping point for toxicity is around that saturable metabolism point. So you might want to just build that in as a possible feature for a user in the future. And then ICE does a very nice job with the different modules. One thing that I heard in the discussion today, but I haven't seen in the tools, it would be nice to automate some of the features so that there's automated processes that are doing the sort of the AI or whatever we want to call it, but the logic behind the scenes that's integrating across modules rather than relying on the user to be able to be a deep user in each of the models and hopefully building the right connections. So I heard Nicole mention some of that already. So I'm hopeful that that will come where ICE will just continue to evolve. So on the compate, so switching gears a little bit, I think it'll, again, very nice work. Happy to see that going on. I think it'd be nice to move beyond the acute tox testing where we can for a variety of reasons. So it'd be nice to understand what the global regulatory community is thinking of this, what would need to be true for accepting in silico predictions for hazard classification of acute toxicity, and making sure that the output of compate is going to be on target with that. There was a brief mention about whether trying to drive to mechanistic interpretation of the data. I think that would be helpful. So whether it's an explicit outcome of the work or whether it's a follow-up activity that can be done since all the data is already collated, I think bringing in different skill sets such as medicinal chemist, toxicologist, biologist to look at some of the data to see, are there chemical features that are either very unique to toxic chemicals, very unique to safe chemicals, that can then be leveraged to help design new, safer chemicals by companies. If we know what to stay away from, we can leverage that. And then just the third thought I had on this and some of the other modeling work that's been done within this space is there was a mention yesterday about a need for additional education or kind of just work at the university level. I have mixed feelings on that. I don't know if we need to change the programs or just give opportunities to students. And I think either during this process or after this process, it would be very unique and empowering opportunity to bring some of these challenges to the universities and allowing people that might not have or routinely have visibility to the calls for action here. So it might be your statisticians, your computer modelers, medicinal chemists, just a variety of skill sets, and give them this challenge as well. And I think this will allow the students and professors to really begin to see some of the real world problem statements that can be solved with their skill sets, because you don't get trained on doing this. I mean, quite frankly, you use the skills and you try to apply them to real world scenarios. I think that's an opportunity that low hanging fruit, but would be great to see. Then just switching lastly to the camera. I like the idea. I think it's going down the right avenue with the questions already posed on the slides. I agree with the watch out of let's not be redundant here and just make a fragmented database. So I'm not going to reiterate any of that more than I just did. But I will say that it seems like there's a natural connection here with the Method Developers Forum that was talked yesterday. I'll leave it to you guys to figure out what that connection is, but it could be that some of this is brought to the Methods Developer Forum for feedback. It might be to try to include some type of dialogue between or within camera and what's happening in the Method Developers Forum to scope out needs. And then also I would say start small. So I think there was a lot of what could be done, but I would start small, see if it works, kind of reiterate where needed. So succeed fast, fail quickly, and then adapt. And then just my last point, which doesn't fit nicely into any of these kind of bullet points here, but just a thought. So there's been a lot of talk the past 2 days about just the in vitro data sets, the computational approaches, but I haven't heard too much about the expert knowledge until just the last comments by Adrian. And I think what lives out in the scientific community that we're not taking advantage of yet is collating that expert knowledge into a common space and then leveraging natural language processing tools to help figure out commonalities. So in some cases, I know within my company of Procter & Gamble, we have expert rules regarding toxicity or regarding different functional groups. And I'm sure others have that either with an assay that they work with or with certain chemistries that they work with. And in isolation, they probably seem like an isolated piece of knowledge. And if you bring them together, they're probably going to seem like disparate, non-connecting thoughts. But just like how Nicole started the meeting yesterday using the natural language processing models to figure out the trends in the different biennial reports, I think that can start to be done with some of these expert rules, expert knowledge, expert judgment that lives within private sector, within government agencies. And then that can help guide new hypothesis testing, new model development, method optimization, and really start to leverage some of that. So that's all I have for now. And again, just thanks and look forward to the continued discussion.
Thanks so much to the SACADM discussants for your excellent feedback. You know, I've been typing furiously and I'm really excited to kind of think about how to put all of this into practice. There was just a couple things that I wanted to highlight that we didn't get a chance to talk about these last 2 days. Those of you who have spent time looking at our website and reading the biennial report know that our portfolio is much broader and much more complex than we have time to go into 2 days, in the 2 days. And so we've, the last several years of SACADM, we've kind of adopted this strategy of doing a deep dive into a couple topics and then having this regular computational tools update. That is by no means the width and breadth of what we're doing. And so just to respond to some of the things that were brought up, just to give you kind of a preview or a little update on stuff that we're working on. It's interesting that you highlighted the Chemical Quest tool. Corey, that's one of the tools that we're, to be honest, the least happy with in ICE, and that we've been talking about, how to really revamp that and how to bring in best practices in both unsupervised and supervised approaches to chemical clustering and categorization and prediction. And I wasn't quite ready for presentation at this meeting, but I'm sure by next year we'll be able to present to you on a tool called MOVES that Kamel's group has been developing that really applies, these sort of novel approaches in endpoint informed and endpoint specific clustering and categorization. So chemical analogs, when you're thinking about something like endocrine disruption or developmental toxicity, are not the same as chemical analogs when you're thinking about something like carcinogenicity or acute toxicity. So having a solely structure-based approach to chemical analog selection is not really something that we rely on internally. We bring in many, many more features like bioactivity descriptors and phys-chem properties in more of a supervised endpoint specific way. And, Kamel's group is building NIME workflows, open source tools to actually implement that. And we were just talking at the ICE planning meeting about how can we pull that into Chemical Quest and really make that a much more modern sort of similarity tool. So really appreciate that, that comment. And then the other comment, sort of this common thread around incorporating not only expert knowledge, but natural language processing and knowledge graph networks. We work closely with resources like BioBricks and the National Science Foundation. NICEDM is a federal partner in an NSF grant with BioBricks on what's called the Proto-OKN, so it's the Prototypical Open Knowledge Network, using large data sets and natural language processing to build knowledge graph networks that are also informed by the expert knowledge that's out there in the scientific literature. And we're really, really excited about that. And I've already written it down as a topic that we probably should dive deeper into for next year's SACADM, because we're making a lot of progress in that area. So just wanted to kind of sneak peek a couple of ongoing activities that we have that are related to the comments that were provided. So thank you.
Great. Thank you.
Thank you. We're going to open it up to other SACADM members to provide discussion. So if you use the raise hand feature, I'm going to start because there's no hands up right now. So my first comment was actually intended for the discussion on DNT, but I was too eager to move on. I guess I forgot myself. So just wanted to mention that I really do appreciate the comparison of time, cost, and animal use for the case study that was presented earlier by Dr. Schaefer. This transparency really showcases the benefit in addition to human health relevance and health protection that's provided by NAM. So I really want to make sure that was mentioned. Okay, and then you're not going to be surprised, but I have a lot of comments. I'm going to try and be quick because we're running low on time. But thank you for that update on ICE. It's such a fantastic resource and really, really great to see the enhanced metrics and coverage. Especially appreciate the updates to the IVIVE tool, and I think this will be extremely helpful for many that are starting to implement risk assessments based on NAMs, as well as comparing to historic data in animals with a nod to validation. Also appreciate that this is a one-stop shop, pulling other data and information into the tool to combine and enhance other data. a great job once again implementing all of the SICATM comments from last year, and that has been noticed and said by other people too. But please do continue to update and expand this tool. It's very helpful for so many different reasons and stakeholders. I also commend the user-friendly nature of the tool. Not being a computational toxicology expert myself, I can attest to this. It is very helpful. And do you have any plans to expand to cover resources for defined mixtures? For example, the ability to predict additivity for finished product formulations, and when that information is available on each ingredient, for example, or if it's known or if it can be predicted. So like utilizing that for those defined mixtures. And then also I wanted to add that any training and information that you have and can have housed online, like a video, that would be really helpful to allow users to learn on their own time and follow along with case study examples. I'm going to pause there because I did ask a question about the defined mixtures.
Sure. Yep. So we do have, several hundred defined mixtures in our database, where we have the acute toxicity 6-pack data set for all of those defined mixtures. What we don't have, what I just made a note of, is we could, implement the GHS mixtures equation as a functionality in ICE, and compare sort of the predicted outputs of that that equation with the data on the actual formulation products themselves.
Yeah, that'd be great. I mean, for me, I personally don't find the defined mixtures that are available they're very helpful, because in order to be able to utilize them, you have to know the exact composition, in my opinion. So, I think it's more helpful to be able to have a tool to kind of do it yourself when you know what the defined mixture that you want to look at is. Okay, so I want to, with regards to the Comp8, I want to commend the team for once again and the awesome amount of work that it must have taken to get the data to the point of use for that tool, not only just for data creation, but for aligning the categories of different agencies and chemical phases. And by the way, what a great idea it was to use another ICE tool in predicting the phase in order to utilize more of that data set. So great job there, and for utilizing your own tools. This will not only be so helpful for predicting single chemicals, but also predicting predictions using the additivity tools like we just talked about, the GTS algorithm. And I appreciate the having the manuscript development as part of the approach, because it helps not only for transparency, but for others to be able to model what you've done for different endpoints. Okay, I really have a ton of things about camera. I'm just going to start by saying I really think that this has huge, massive, massive potential. And I think that we should have a workshop on this point, and I am happy to volunteer to be part of that. Just in case we don't get to my comments, I'm going to stop there and let others speak, and then I'll circle back if there is some time. Sally, go ahead.
Yeah, thank you, Sally Thompson-Ertani. Sorry, I don't think I've always introduced myself, but I just have two kind of quick questions. One is, we're looking at the user groups and thinking about, I don't currently work in industry, but I also did previously, and the people that were making the decision about what needed to be done was the regulatory affairs group. And how are we encouraging them to utilize these things? Are we able to? Maybe they're doing great. I'm just kind of curious of what that looks like. But I also know they're really looking at the global market, and that's ultimately driving a lot of their decisions. And I know that we talked a little bit about this, I think even last year, but are there additional things, or just looking at the United States as trying to be a leader and then, making these resources available, is that going to help with this? So those are kind of my questions.
Yeah, I mean, to your point, I have attended conferences of, that were largely regulatory affairs folks from industry and done hands-on trainings, like half-day trainings of ICE, and gotten really excellent reception from those. So that is definitely a user community that we are, at least to a certain extent, engaged with.
Jonathan.
Go ahead, John.
Yeah, John Gordon, CPSC. Yeah, that's a really good point to make, because a lot of people, just out of, I hate to say, but ignorance, don't really know anything about the science and don't really want to go down that road of, I don't know how to use this data, so I'm not going to. So a lot of that is actually on us in regulatory community, making sure that we're clear on what we accept, how we accept, and when we will accept things. So there's a whole lot of that that finger needs to point at me. And we need to make sure we're clear about how to do things, so that when people do go to the CPSC webpage and say, hey, I want to use Crocedex or any of the skin sensitization ones, how do I do that? You can go there, you can find the information, and you can use it properly in your regulatory program, so that you can do this. So there's a whole lot of clarity that needs to be done from the Fed's point of view on this. So that's a step that we can take as a group as to how to do this. There's other steps as well. But one of the things that we definitely need to do is be very clear on what we do, how we do it, how we will accept it, and what data we will accept. And the camera, I think, is going to be a fantastic tool for that, because you can click on CPSC and say, hey, what does it'll have much information on CPSC. Maybe it'll direct you to our website, how we do things. Maybe it'll have all of our memos on the methods that we've reviewed and exactly what we want to see from each of those methods, so that you can put that into your regulatory scheme.
Okay, Nathan, go ahead.
Thank you. Nathan Price, Buck Institute in Thorne and SCATO member. So, yeah, super interesting session. My question's about the, I guess just related to ICE and its use. I'm thinking a little bit like, some of the things that Misty was going like as a user. So I'm kind of imagining like me or someone like me coming, to be a user of this. So it's got a ton of really interesting information in there, obviously on acute toxicity, it's carcinogenic, a whole bunch of different things. So is it set up in such a way that if I have, let's say, like a metabolome out of blood, or if someone had or a sample of soil or something like that, where you have just like some list of chemicals, right? Is it easy to upload that into this? It's robust, all these different kind of chemicals, and you can hit that back. Is that hittable by like an API if you set these kind of things? Just kind of walk me through a little on how that interface looks. Do you need a human in the loop? Can you hit this multiple times? How does it interface back to the end user, I guess?
Yeah, I mean, you can hit our API with a list of chemicals and get all the data in our database back. That's absolutely doable. We're also thinking about how to extend the API functionality such that you could automate calling some of our workflows too. So that you could programmatically interact with things like the IBIBE tool or the Chemical Quest tool or the, chemical characterization tool. But at a minimum, all of the information in our database, which includes things like exposure data and, all the toxicological data, all the predictive properties and tox endpoints, you can pull all of that programmatically through the API.
Yeah, that's great. Are there limits on, I guess, if other groups come in and do that? If someone hit that very repeatedly or something, are there limits to that, or do people if a big company were coming to do that or something?
Yes, I can't remember there's I would need my developers on here to answer that question, but there are batch limits, but we were talking about that a few weeks back. I don't know, Kamel, do you know anything additional on that?
Yeah, I don't know exactly, but you're right. Yeah, there's the limit on the batches.
But we're looking at doing things like, having not a timeout, but basically say like, okay, you've put in this query, it will be ready in X amount of time and it will get emailed to you or something like that, so people aren't just sitting waiting. Right. But we did just get a new box, a new server, and it's like way nicer and way faster. So that's exciting.
Congratulations.
What I just got back from the team, this is Emily Renke, and it's NICEDM, and they're saying it's approximately 50, but it depends on the tool. So there's a few that can go up, I think, to like 1,000.
Yeah, but that's through the user interface.
So that's the user.
That's
for the API.
They're much larger.
Yeah, they're not
API. Yeah.
But just one quick follow-up. Are there like really significant major users of this then? You showed some usage data early on that seemed quite long. Is there this sort of dominated by a few places that do a lot of this, or is it pretty spread out with a lot of smaller users?
I mean, it's something that we're really trying to figure out better tools to answer. So we have like Google Analytics reports that we get every month that allow us to see like where the users are coming from, but that doesn't associate them with like agencies or universities or companies or anything like that. We just get IP addresses that tell us like where geographically they're located. And so what I alluded to before with wanting to create a better sort of literature search strategy that is not just my LinkedIn feed to tell us when, papers are published that rely on ICE as a foundational tool for what the work that they're doing, I think is something that could really help to track that. But I've been really impressed to see that, there's users and regulatory authorities in the EU, in Korea. It's not just in the US. And then research institutions as well are increasingly starting to use it to, their workflow.
As that sort of report comes back, so you hit ICE, right, with some set of chemicals, it's going to come back and it will tell you, here are chemicals you should worry about for acute toxicity, here are things you should worry about as carcinogens, here, and all that. And then it just comes back sort of as lists. Or you were saying that even some of the workflows
you get the data.
You could maybe integrate in or whatever.
Well, so I think one of the biggest applications that we've seen of ICE is, so there's two really, people use ICE, all the different tools, but I think the two biggest tools that get used are the search function, to interact with the data sets, download the data sets, join different data sets together in a way that you don't know how to do unless you're a programmer and you're working with flat files of data. The data portion of it is highly used. And then the IVIVE and the PBPK workflows to actually take in vitro data, whether it's from the ICE database or whether it's user-generated in vitro data, and then apply those reverse dosimetry models to say, okay, what would I have to be exposed to attain those internal concentrations? And then compare that to either in vivo data from animal studies, again, from the ICE database, or uploaded by the user, or human biomonitoring data, exposure information. There was a really beautiful paper that I was privileged to contribute to that came out of a group in Brazil, where they looked at exposed agricultural farm workers that had really high exposure to triazole fungicides, and they used ICE as like the tool to take the human biomonitoring data and relate it to in vitro activity concentrations, and found that the targets that they were seeing perturbed in all the biomarkers that they were looking at in the ag farm workers. So nuclear hormone receptor pathways, metabolic pathways, oxidative stress, genotoxicity pathways matched almost exactly the in vitro perturbations. And when you did that IVIVE, it was happening at the same exposure levels that they were measuring in that exposed cohort of humans. And so without that tool to facilitate, making those comparisons and having that computational workflow as its basis, I don't know that work would have happened or that paper would have come out, and then we wouldn't have this weight of evidence that they can now go back to regulatory authorities in Brazil and use to make a case for better risk management, for better support around personal protective equipment, availability, and, actual utility.
That's great. Thank you.
Dr. Silveira, go ahead.
Silveira, Indiana University. Along the lines of outreach and collaboration, I wonder if it is a plan or maybe should we consider to reach out to program directors of training programs or even accreditation bodies to ensure that the type of tools and training are incorporated in curricula and prepare the next generation. And also, I wonder if, I guess I got a little frustrated when I saw the map and which countries are using this. And I wonder if there's an opportunity to connect with organizations that do work or fund work, like GATE, like in parts of the world where certainly, like the example you gave with Brazilian workers, like there are more vulnerabilities based on the type of exposures there are and the regulation, and maybe there could be some effort to expand the globe beyond what's already.
So that is something that is really near and dear to my heart as well, because it is frustrating to me to see tools like these, like the lack of awareness of tools like these that could help support the people who are disproportionately exposed to environmental chemicals, not being taken up in the areas of the world where that exposure is happening. And that's simply because of, lack of awareness, lack of training, and funding. So one thing that I have personally started to challenge myself to do is to leverage my conversational fluency in Spanish and try and expand that to more professional fluency. And I've started to give presentations to congresses in Latin America in Spanish and provide training on these tools. I have one tomorrow that I'm kind of stressed out about because I'm not ready for it yet, but I will be. It's being hosted by a group in Colombia, and I'll be talking about ICE and specifically about the IVIVE tool and how it can be applied to real-world data sets. And I will say that the last one I did of these that was hosted by a group in Argentina, it was webcast. I didn't know until after the fact how many people were online, and there were 700 people tuned in live to the webcast, which, when we run an event, we think it's a success if we have like 200 or 300 people online. And they've told me that, twice as many have viewed the recording afterwards. And so we also, like for the exam strategic roadmap, we translated it into 5 different languages. We're doing the exact same thing right now with the validation guidance document. So translating it into like Spanish, French, Chinese, Portuguese, Japanese, Korean, trying to hit different parts of the world and actually like make it more accessible in a global way.
Dr. Baines?
Antonio Baines, Associate Professor, NC Central, Cicada member. So kind of on the same regard to that, this information needs to get to our undergraduates and graduate toxicology programs. Have you thought about how to get that information? Because again, this is like kind of like how to press, real-time techniques to help, push toxicology to the next level. But I'm not sure if it's getting to those students. And so just want to hear your thoughts of how you and your team want to try to get this information out there to various especially grad programs. Undergrads might be a little too much, but definitely graduate programs in toxicology. Just want to hear your thoughts about that, just in terms of, again, as we've been seeing, getting this information to the next generation. That's going to help push this NAMS story forward.
Yeah, so I mean, that's super important. We do that by direct engagement with individual university programs, UNC Chapel Hill, Johns Hopkins, NC State, Texas A&M, Yale, School of Public Health. Those are just the ones off the top of my head that I am, specifically engaged with, but would love to expand that portfolio. But then I think we also really try to do it through like the SOT annual meeting, Society of Toxicology annual meeting, to reach a broader swath of trainees and graduate students and early career researchers. But can always do better, can always expand that, and, would really welcome connections and opportunities from our esteemed SACADM members and any of your networks that you would like to put us in touch with. That would be wonderful.
Okay, I am not seeing any hands up, and we're running a bit late, so I am going to take the opportunity to move into the concluding remarks section. I am just going to say that it's been a pleasure to be the chair, and I have really, really enjoyed participating in SACADM. I hope to come as a public participant in future meetings where you don't have to listen to what I say. And I am going to pass it on to Dr. Pastel to make some concluding remarks.
Yes, thank you. So I really want to thank everyone for a very innovative and thought-provoking meeting. It was a lot of content, and it was like drinking from the fire hose for a little while, but I think and I am a neurotoxicologist, so, I just commend all the speakers, and especially Nicole, for the really terrific content. I know that we all learned a lot, and I especially want to thank the committee for their input and feedback on these emerging technologies and approaches. There were a lot of ideas and comments that emerged that many of us are already discussing, and we're going to take back with us and reflect on as we advance technologies. And I want to end by reminding us, as we sit here in this terrific space at the National Institutes of Health, that the ultimate goal here is health protection. So I'm really pleased that the discussion wrapped up with that in mind, and not just with vulnerable communities in the US, but globally. And so I really appreciate that that's where we landed and where we concluded today. I think that's incredibly important. There's been a lot of talk over the past couple of days about the users, but ultimately the most significant stakeholder is the public, and for them, the proof is in the pudding. So I appreciated the question of how are people using these and how are we influencing decisions? And so I'm really excited to see, as these technologies advance, how they impact decision making. And I'm confident that these technologies will live up to the promise that they will enable more health protective decisions. And I think the next time this committee meets, there may be some really good examples of that, which is really exciting. And hopefully we'll get there. And I really appreciate the effort to make these tools easy to use across different disciplines, so that chemists and engineers and other groups are enabled to develop safer technologies. Humans are an incredibly innovative species, and we're certainly capable of that. And I really commend this group for developing user-friendly tools that will help enable that. So I really appreciate all the comments and the diversity of thought over the past 2 days, and commend all of you for your work. And I thank the chair for her terrific job of keeping us all on time and focused. I'm sure she's excited to be past chair, which is the best job ever, right? And to Nicole for organizing a really informative and productive meeting, and for everyone online for their contributions. Thank you very much.
This has been a great opportunity. I'm definitely sad to be rotating off. I am going to pass it to Dr. Kleinschreier for her closing remarks.
Thank you. So we've had so much amazing and robust scientific discussion that I'm not going to dive into any specific aspect of it. I have 5 pages of notes. So I'm really excited and just so grateful to all of the SACADM members for taking your time and providing such thoughtful and actionable comments. That's one of the things I love about this committee, is that it's not just, kudos. It's really, here are things that I think you could do to improve and challenge us in many ways to continue to be better. So thank you all. Thank you especially to the State Academy members who are rotating off this year for your service over the last 4 or 5 years. Especially thank you to Dr. Page for her leadership in the chair position and all of her many contributions, of which I expect to get several more paragraphs in writing. I want to thank all of our speakers, not only our NICEDM speakers and initiative speakers, but our colleagues from the NIH Office of Strategic Coordination, Dean Poughkeepsie, the Foundation for NIH, EFSA, EPA, FDA, and our NGO partners. Really need to thank Initiative. The incredible work that NICEDM does in support of ICFAM would never be possible without the Initiative team. They are the lifeblood of the NICEDM group, and so a huge thank you to them. In particular, thank you to Emily, Stephen, and Kathy, who have worked tirelessly to make this meeting a success. Also really want to call out the SIOM subcontract support. They provide amazing support on the ICE side, the database, the software development, and lots of bioinformatics support as well. Thank you to all of our ICFAM representatives who took time to be here today and for dialing in online. Thank you to our public commenters for your thoughtful feedback, and all of the attendees who came and listened in and provided feedback. Thank you very much to Mary Wolf, Mulaney Brownlow, Ella Darden, Gail Burnaby, and Robin Guy for actually making this meeting happen. So they would not happen without them. And last and most importantly, thank you to our AV team for your incredible technical support. You are always amazing and a joy to work with. So thanks to everybody, and thanks to anybody that I forgot to mention. Catch all.
Yep, and Dr. Brownlow, for your closing remarks.
Thank you. Thanks, everybody. So I will not repeat everybody that Nicole just mentioned, but thanks everyone for your active participation and engagement. Thanks for everybody who's in the room, everybody who joined us online. And just as a reminder, we will post slides and videos of this meeting on the website once they meet government requirements, and we will also post the minutes for the meeting after they've been reviewed and approved by our chair and meet government accessibility requirements as well. So thanks, everybody.
Thank you. At this time, we usually do open it up to other SACATA members for concluding remarks, but in the interest of time, we are over time. If you have any additional concluding remarks that you would like to submit in writing, Nicole is happy to receive those. And with that, we will adjourn the meeting. Thank you very much.